Bonjour à tous. Donc juste pour information, on est maintenant en streaming sur YouTube. Donc la présentation est rediffusée sur YouTube avec les slides, si jamais.
Est-ce qu'on peut flotter mon visage?
Vous m'entendez bien?
Ouais.
Parfait. Just for your information, we're just waiting for the members of the jury right now. François allait aller chercher.
commencer. Le jury est au complet, donc on peut commencer. Donc, Monsieur Jonathan Cottet, nous sommes réunis aujourd'hui pour examiner votre travail en vue de l'obtention du doctorat de l'Université de Lyon dans le domaine de l'électronique, l'électrotechnique et l'automatique. Voilà. Donc le jury qui est réuni aujourd'hui est un jury hautement international, puisqu'il rassemble le professeur Vandenberg de l'Université de Twente, qui est également un de vos rapporteurs, le professeur Olivier Français, à ma gauche, de l'Essier, votre second rapporteur, également le professeur Ramos de l'Université de Séville, moi-même, qui sont donc la présidente de ce jury et qui suis chercheur CNRS au LAS à Toulouse, et vos encadrants, François Buret, professeur des universités à l'École centrale de Lyon, Philippe Renaud, professeur à l'École polytechnique fédérale de Lausanne, et Marie Frénéat-Robin, maître de conférence à l'université Claude Bernard, ici à Lyon. Donc vous avez, comme il se doit, 40 ou 45 minutes pour nous présenter votre travail, après quoi nous aurons certainement quelques questions à vous poser. Voilà, vous avez la parole.
Thank you, Anne-Marie. I will do the presentation in English for the sake of understanding. So, thank you everyone for coming to my PhD defense about my PhD thesis work, which is on the development of microsystems for the controlled formation of cell aggregates by dielectrophoresis. It's a work that has been carried out between two labs, one located at École Centrale de Lyon, the Ampère Laboratory, in the bioengineering department, more precisely in the biomicrosystems, bioelectrochemistry and bioelectromagnetism team, under the direction of Professor François Buret and Professor Marie Frénéat-Robin, and also in another lab at EPFL in Switzerland, in the Laboratory of Microsystems 4, under the direction of Professor Philippe Renaud. As I said, my PhD thesis is about the development of microsystems for the controlled formation of cell aggregates by dielectrophoresis. But the question is, why cell aggregates? If we look in the human body, it's composed of different tissue of different natures, and it's constantly exposed to different electromagnetic fields. It's quite difficult to model it directly in the body, and while sometimes the electric field can be detrimental for the body, sometimes it can be beneficial, like in electrochemotherapy, which I will introduce a bit later in the presentation. But understanding it directly at the human scale, at the body scale, is very difficult. So what has been done is usually work at the single cell level, when you have a single cell and you expose it to an electric field. The problem is that the results we get for a single cell, usually in suspension in a liquid, are quite different than what we could obtain directly for the body. So we're in need of an interim model to bridge this scale gap between single cell and the tissue, and we propose to have cell aggregates where we can control the size and the composition of the aggregate to be able to go from the single cell to the tissue scale. And our work is about bringing this intermediary model. More precisely, the work is kind of divided in three parts. First, we want to work on the creation of the aggregates by dielectrophoresis, a concept that I will introduce a bit later. So we want to be able to bring cells together to create cell aggregates of controlled size. Then we want to be able to study those aggregates directly using an electric field by what we call impedance spectroscopy. After having these aggregates that we have characterized, the final goal is to be able to electroporate those aggregates and to be able to see how the electroporation parameters should be tuned when we change the size of the aggregates and its properties. But in this large project, we decided to focus on the first step to be able to control and to create those aggregates and to be able to have tools to study them. And that's what we bring in my PhD thesis. So the presentation is divided in 9 parts. First, I will introduce you all the theoretical concepts. Then I will go for a brief state of the art on what has been done in the lab and internationally. Before going for some modeling with our software, MyDEP. I will then present you what is the design of our microfluidic chip and the simulation that have been realized before introducing the fabrication of those chips and all the experimental results. After we have those aggregates, to study them, I will introduce the concept of impedance spectroscopy and impedance sensing and the work we have done related to this field and how we combine it with dielectrophoresis. I will after give you a short conclusion and an outlook of this work. So first, theoretical concept. As I say, we want to create cell aggregates by dielectrophoresis. So what is dielectrophoresis? So for the definition, dielectrophoresis is a phenomenon in which a force is exerted on a dielectric particle when it is exposed to a non-uniform electric field. And if we look at the force applied on a dielectric particle when it is suspended in a medium, we have the following equation. It's divided in three terms. The first term is about the geometry, more precisely the volume of the cell we want to interact with. The second term is about the electrical properties of both the cell we want to work with and the medium, the suspended medium. And the last term is about the electric field, more precisely the voltage we will apply and the magnitude and also the configuration of the electrode. Now if we look closer in the electrical properties, what we see is the Clausius-Mozotti factor. And this term reflects the complex permittivity of both the particle and the medium. And this complex permittivity is linked to the permittivity, the electric conductivity of both the particle and the medium, and also the frequency at which we are working with the electric field. Macroscopically, we have two effects. Either the real part of this factor is positive, and in this case, we will attract the particle in the high field region. That's called positive dielectrophoresis, also called PDEP. Or, second possibility, the real part of the Clausius-Mozotti factor is negative, and in this case, we will repel the particle from the high field region, and they will go to the low field region, in this case in blue, which is called negative dielectrophoresis, also called NDEP. So if you want to go for a more physical explanation, what is happening at the particle level? If we have two electrodes, one positive electrode and one negative electrode, and we have a dielectric particle in suspension, we apply the electric field. What we'll have is a small displacement of the charges inside the particle. In this case, the positive charge will face the negative electrode, and the negative charge will face the positive electrode. Now I say that the particle is less polarizable than the medium, which means that we will have more charges coming from the medium than we have moved inside the cell. So in this case, we have positive charge on the left and negative charges on the right. And if we have a closer look, we see that now the right part is slightly more negative and the left part is slightly more positive. So we have created a dipole. And in this case, we will have a dipolar moment inside the particle, and we'll have the forces applied by each electrode on each side of the particle. In this case, we have an equilibrium between the forces, so we don't have any net force because we don't have a gradient of the square of the electric field. But now if we modify this configuration, what we obtain is a gradient of the square of the electric field, and in this case, we will have a difference in the forces applied on the particle, and we will induce a net force and a particle movement. That's called, in this case, negative dielectrophoresis. The particle will go in the low field region. So now, what can we play with? We can't really modify the cell, but we can play with the medium and with the frequency of the electric field. So in this case, I'm presenting for a Jurkat cell, which is, in fact, a cell composed of a cytoplasm and a membrane. And if we look at different electric conductivity of the medium, we can see that the response in frequency is different. For example, if we look at a medium with 10 millisiemens per meter of conductivity, what we'll have is the following curve in dark blue. And we see that there is an interesting frequency that is here, which is a crossover frequency, which means that at roughly 35 kilohertz, we'll have a transition for a regime from the negative dielectrophoresis regime to the positive dielectrophoretic regime. So that's how we can play a bit with the parameters. This is for the creation of the aggregate, where we are using dielectrophoresis. The next part is for the impedance, for sensing what we have created, and it's linked to bioimpedance. The concept is the following. We have two electrodes, and we apply an electric field, a voltage, between those two electrodes. We will have a current flowing from one electrode to the other. If we have a particle between those two electrodes, the value of the current will be modified. And if we increase the frequency of the current, we'll see that the current now will enter into the cell and be able to probe what is inside. And the reason is the electrical model, because at low frequency, we will only probe what is happening in the medium, and then by increasing the frequency, we will short-circuit the membrane, the capacitor, which is equivalent, and we will start to probe both the resistivity of the outside and the resistivity of the cytoplasm, which we can see in the bot plot. Low frequency only the exterior of the cell, and basically its size, and high frequency, both resistor in parallel, extracellular and intracellular. The last concept now is about electroporation. So what is electroporation? In this case, we have a cell and we apply a very short, very intense electric pulse. What happens is that it will create a pulse when we apply this electric pulse. And right when we stop it, the pulse will close. But it will have induced defect in the cell membrane. And those defects will collect it to reunite each other, and they will open larger pores in the cell. And if we do it correctly, the pores will be able to close on themselves after some time. Now, if we use it in the framework of electrochemotherapy, it's the same principle, but in this case, we add a cytotoxic drug just outside the cell, and right after we apply this electric pulse, the drug will diffuse inside the cell. And if it's a cytotoxic drug, in this case, for example, bleomycin, what happens, it will induce defects for the cell in division, and it will kill them. So only the cells in division will be killed, while the drug will enter in all the cells that are permeable. This concept is usually used for transfection, for cell transfection, which means that we have a little cuvette made out of two electrodes, we have a liquid and cells in suspension. We apply a certain value of the electric field and we enable materials to enter into the cell. Now if we want to use it for electrochemotherapy, it means we want to use it with patient, which means we need to adapt this little cuvette to the human body. We can't directly insert it in the tumor. So some work has been done to try to find a design that is more suitable to use it directly in the human body, like this one, composed of many electrodes. As I said, the problem is we need this intermediate model to work, because between the single cell in suspension and the human body, there is a big scale difference. And this technique is not only an academic technique, it's a clinical technique. And just for your information, those are the centers where electrochemotherapy is used in Europe. And for example, there is a center right here in Lyon and another one in Geneva. So, that's all for the theoretical concept. Now I will go briefly over the state of the art. So the first work I want to mention is the work done in the Ampere laboratory, and especially by Samia Menad in her PhD thesis. In her PhD thesis, what she did is she used electrodes patterned at the bottom of the microchannel, which are insulated partially with PDMS. So she has a quadrupolar structure, and she was able to create HEK cell aggregates, which are human embryonic kidney cells, of a controlled size. The second work I want to mention is the work done in the lab on the electroporation. There was quite a lot of work on modeling the electroporation at the single cell level, and now the current project is to try to move this understanding of the cell level up to the tissue level. Just as a conclusion of this work, performed in Lyon, mostly in the thesis of Samia Menad, I want to say that it was possible to have a collective fabrication of cell aggregates of regular size, and second point, that the use of an external force increased the interaction to be able to create permanent and viable cell aggregates after 10 to 15 minutes of exposure to an electric field. The second part is the work performed at EPFL in the group of Philippe Renaud, and especially I want to mention the thesis of Nicolas Demierre, where he developed the concept of the liquid electrode. In this case, you have coplanar electrode patterned at the bottom of a microchannel, with a little recess from the central channel. What happens in this case is that electric field will be homogeneous when it reaches the main channel, which means we kind of have an equivalent from this coplanar design to a 3D equivalent with electrodes that could be virtually seen as two vertical electrodes. And this design has been mostly used by Nicolas Demière to both focus cells and also to perform some sorting. The second work I want to mention is the PhD thesis of Marjan Shaker. First, what she did is she retook this configuration of the liquid electrode and she performed additional simulation, and she showed that not only by having one side at one potential and the other side at another potential, she was able to laterally center the cell, but there was also in the simulation visible an effect on the lifting, on a kind of vertical centering. And she was able to combine this effect for first centering cells, and then afterward using the impedance principle to sense the anisotropy of the cells. So now if we look at this design, as I said, you go from coplanar electrode to a 3D equivalent electrode. That is a vertical equivalent electrode. Now if we go look more internationally at the state of the art, I want to mention the work done in the Faux group by Dr. Jaeger, where he used electrode patterned at the top and at the bottom of a microchannel. At the beginning to center cells in the microchannel, and afterwards with a configuration composed of 8 electrodes to trap cells directly in a cage. So in this picture, for example, he was able to trap one single yeast cell by DEP, and while keeping the DEP active, he let it for a long time until it divided for more than 22 hours, and it was touching at some point the top and the bottom of the microchannel. The second work I want to mention is the work done by Dr. Bögel, where he worked both on electroporation and impedance spectroscopy. In this case, he used 2 electrodes to sense the impedance of the cell while it was passing, and then he used another set of electrodes to perform the electroporation of the cell. And he used a pressure control to go back and forth in the microchannel. So the cell is sensed, then electroporated, then go back and forth with this configuration. So with this state of the art, I want to show you that it's possible to create cell aggregate by dielectrophoresis, while we can't strictly control all the properties of the aggregate. And it's possible also to use impedance spectroscopy and electroporation on the same chip. So now if I go back to my PhD thesis work, it's the development of microsystems for the control formation of cell aggregate by dielectrophoresis, and the characterization by impedance spectroscopy in order to use them as tumor model in the framework of the study of electroporation. And as I mentioned earlier, what we decided to do is, before trying any electroporation experiment, we decided that the most important part was to be able to create cell aggregate in a reliable way of control size. Which means that we need a different set of requirements. First, we want to be able to trap cells under flow condition. And we want to accept new cells to join the aggregate we are creating. We want the size of the trapping area to fit the size of the aggregate we want to create, roughly 50 micrometer by 50 micrometer by 50 micrometer. We want to be able to combine different cell types inside the chip. We want to have a stable trapping, even if we have some flow variations. We want to have a simple fabrication process. We want the cell to be still viable after some time. And we want to integrate in this chip an impedance sensor for both single cell and cell aggregates. So now if we go back to the presentation. Before starting any manipulation in the lab, first we need to understand how it is possible to trap cells by dielectrophoresis and how different cell type will be in. So what we did is we created a software, which is an interface, mostly with Olivier Fabrec from École Centrale de Lyon. And it's now a work that is published in the Biophysical Journal. So the interface is the following. We are able in this interface to set the parameters of the medium and to choose the cell model we want to work with, directly with all the characteristics of the cell. And we are able to generate all the graphs that could be needed mostly for dielectrophoresis, but also for impedance, because we can generate the Clausius-Mozotti factor graph, both real part and imaginary part, and also graphs related to the complex permittivity, to the relative permittivity, to the electric conductivity, and to the crossover. And directly in the interface, we can analyze the graph we have generated, and we can mix the curve coming from different cell models. So the cell models that have been implemented are the following. We have the homogeneous particle, for example, typically a polystyrene bead, a single shell, which could be a cell with its surrounding membrane, a two-shell model for a cell that could be more complex in the behavior of its membrane, like a yeast cell, and a three-shell model. In this case, it's when the nucleus has a significant size inside the cell. And we did this work for spherical particles, but also for ellipsoidal particles, because not all cells are spherical. For example, bacteria are sometimes not spherical. And while we have all those models, we have more than 78 values that are coming from the literature that I compiled in the database, where we can directly integrate it in the interface, which means that if the user wants to understand what would be the behavior of a certain set of cells, let's say, for example, the HEK cell that Samia used in her PhD, it's visible in the interface, and all the references linked to this set of data are directly accessible. For example, we can see who wrote the article, where it was published, and directly access the journal. So with this work, we have a database with values from the literature. We have references that are accessible right through the interface. We can also create a new set of data that can be submitted directly online to for us to look. And the database can be updated remotely. Second thing is the sweep. To be able to better understand what is happening for a cell when we are working in a medium, we allowed to perform a sweep on all the parameters of both the medium and the cell of the different model. In this case, for example, if we want to work with HEK cell and we don't know what should be the electric conductivity of the medium to perform some trapping, we can perform a sweep directly, in this case from 1 millisiemens per meter to 1.6 Siemens per meter, corresponding to a more culture medium. And we see that in this case, we go from having almost no negative dielectrophoresis to having a large portion where we can have negative dielectrophoresis. And while this conductivity can be interesting because we are directly working with a cell medium, the problem is we can have too much Joule heating, so it can be interesting to reduce this electric conductivity. We can also create another set of graphs. For example, if we want to study the cell more with the impedance concept, it's interesting to be able to see what would be the behavior of the cell alone, like here in those curves in blue, of the medium alone in orange, and how the cells in suspension as a specific volume fraction will modify the properties we are seeing, which is the green curve. And we have this for the complex permittivity, for the relative permittivity, and also for the electrical conductivity. And all those results are compiled in one software, as I said, MyDEP, that is available freely online for the DEP community and all the users that may want to use this software. And importantly, we have a link to the article, and all the equations that have been implemented in MyDEP are given in the supplementary material of the article we published in the Biophysical Journal. So that's all for the modeling part. Now that we know how to model the cell, the question is, how are we going to trap them? And that's the following part. So first we started with the software, MyDEP, and we tried for our HEK cell line, and we said we want to have, let's say, 10 times diluted PBS or cell medium. We see that we have the red curve, which is a real part of the Clausius-Mozotti factor. By looking at the range of frequency we could work with, we saw that working at 10 kHz, we could have the maximum negative DEP force. So with the interface, we were able to choose both the conductivity of the cell medium and the frequency at which we are working. Now we need to find a design, and we choose the following test design composed of 8 electrodes disposed in a circle. Those electrodes are based on the liquid electrode concept, which means they are, in this case, 50 microns away from the trapping chamber. And the question was, what would be the optimal configuration of those electrodes to be able to trap cells directly in flow? So I performed a certain set of COMSOL simulation, multiphysics finite element method simulation, and by testing different configuration of electrodes, 2, 3, 4, 5, or even 7 electrodes, I saw that some cases were allowing the trapping and some were not allowing the trapping. And then by looking at in which case the trapping force will be maximal, which means that in this case we will be able to trap cells for sure, I found that the best configuration for trapping cells in flow was using those 3 electrodes. And if we go now more in details in what the simulation are composed of, so the first part we are looking at the microfluidics, so at the fluid flow directly in the microchannel. So we see in this case that we have a certain fluid flow, and then the fluid will take all the volume, so the speed will reduce, and then will increase. The second set of simulation is about the dielectrophoresis, which is the gradient of the square of the electric field, which is not something easy to figure out without simulation. So we see in this case that combining those 2 forces, the cell would be trapped here at roughly 24 micrometers, which means that we will have an equilibrium between the drag force in green and the DP force in blue. And this position would be a stable equilibrium. So now we have some simulation, we have a design, now we need to be able to fabricate this design. And that's the following part, and that's also a work that we published in Biomicrofluidics last year. So this is how our microfluidic chip looks like. I have a set of microfluidic chips that I would like you to pass through the racks, please. So this is how our microfluidic chip looks like. We have titanium and platinum electrode deposited on glass, and on top of that, we have a microfluidic microchannel directly in PDMS. It's made first by taking a glass wafer where we sputter titanium and platinum electrodes. We use photoresist, we use direct writing, and after what we developed it. So we have some electrodes that are protected, and now we can use ion beam etching to basically only have the electrode part on glass. So we have the glass with the electrode, for the first part. Now we need the microfluidics, and that's the second part of the fabrication. So in this case, we have a microfluidic channel based on using first a silicon insulator wafer, where we spin coat a photoresist. We use direct writing to protect the part we want, where we want to have our mold, and then we use dry silicon etching, resist stripping, and then we have a mold where we can perform a salinization, and then add some polymer, in this case PDMS, that we would pour on top of this mold and unmold it. So it means in this case now we have 2 parts. We have the glass with the electrode on one side and the microfluidic part in polymer on the other side. And we need to be able to align them together and to bond them. So it can seem easy. We have, imagine, this flat PDMS at the bottom, the electrode pattern on glass, and we want to align it. The problem is if we want to align it by hand, it's quite challenging because the microfluidic channel we are using are 50 micrometers. But that's a theoretical concept, and we can perform some alignment. But now if we really fabricate PDMS, it's never almost completely flat. So it's more generally the second case, whereas the PDMS is a bit tilted. So if we want to align, the problem is, in this case, it will be partially misaligned. So the first constraint to be able to perform this alignment is first to have a flat PDMS and parallel surfaces between the top and the bottom. And for that, what I created is an enormous sarcophagus where we can use directly a wafer using and maintain in UV tape. We have many pieces in PMMA that are used, in fact, to be able to pour PDMS vertically directly in our mold, and then we can put it horizontally in the oven for curing, and afterwards we can re-separate all the chip together. So now we have the microfluidic chip. The question is, how are we going to align it? And we looked in the clean room and we said, what is the best machine we could use to perform an alignment? And the answer was a mask aligner. So what we did is, in fact, we hacked the mask aligner, and by using the first substrate to maintain the microfluidic part, we changed the chuck by using a steel chuck with magnets, and we were able to reliably position our chip. So, which means that now we have the microfluidic part on top, the electrode on the bottom. We can perform a pre-alignment, put everything in the plasma, bring back everything into configuration, and then put the two into contact and perform the bonding. So for example, you see here, this is the result, and at the end you have the microfluidic part and the electrode that is re-separated after bonding. And this is the result, and just to show you, those are 50 micron microchannel, and this is 60 micrometer electrode. So the alignment we can perform is in the order of the micrometer. So now we have our microfluidic chip. The next part is to test it. And that's a work I performed with two students mostly, Alexandre Kéran and Soufiane Nasli. So we have the microfluidic chip. We know what frequency we should use. So we went to the lab and we tried first with beads, polystyrene beads. And what we saw, using a free electrode configuration, as I presented earlier, we were able to trap polystyrene beads, first one polystyrene bead, and then additional beads, little by little. In this case, we went to 1, 2, 4, 12. And what we saw is, little by little, as the beads were kind of forming a cluster, the cluster was getting centered in the microchannel. So in this case, we are able to create a cluster of polystyrene beads. The question is, can we use now the same concept with cells? Is it going to work? Are we going to trap cells? And the answer is the following. We are using a cheeky cell, and we are first to passivate the microchannel to prevent the addition of the cells. Then we have to control the medium conductivity to reduce Joule heating. And in this case, we also have to correct the osmolarity of the cell, of the medium, to have physiological condition, let's say 300 milliosmoles. And using our configuration with the free electrode supplied with voltage, what we see is the following video. In this case, we have those free electrodes supplied with voltage, and we have HEK cells that are coming from one single outlet, and that could escape using one single outlet. In this case, you see that the cells are getting trapped directly in flow, and that they are forming little by little a cluster composed of cells. The more we wait, the more we have cells joining the cluster, and at some point we have, let's say, the aggregate we want with a certain number of cells. The question was, okay, we can form aggregates, but what about the integrity of the cell? So we continued for the experiment for a long time, and we added trypan blue directly in the solution. And what we saw is, for smaller aggregates, we couldn't see any coloration, but when we increase the size of the aggregate, you can see that in this case, roughly 30 cells, the aggregates start to be pushed more to the outlet, and then in this case, cells starting to be permeable, and we could see that the trypan blue entered in the cells. And once we have an enormous number of cells, we could see that the cells were really going too much in the high field region and exposed to a too high temperature. So finally, I want to say that in this case, we were able, with a free electrode layout, to perform the trapping using free electrode. Large aggregates, unfortunately, are not stable with this configuration. They can be also exposed to a high field region of the electric field. The fluid can also reaccelerate one really close to the trapping position, which makes this position a bit unstable. And also one significant limitation is with this design, once we have the number of cells we want, then we still have cells coming little by little. So in this case, what we proposed to do was to move electrode farther away from the trapping zone, to modify the fluidic layout to increase the trapping, and to reduce the flow and the dP force once we have reached the number of cells we want in our cell aggregate. And that's what we did. And you can see in this case, the microfluidic chamber for trapping is larger. The reacceleration of the fluid will be no more inside the trapping chamber, but after. And we are also using auxiliary channels to still reduce the speed of the particle while they enter in the trapping chamber. So we tested this design in simulation, and we saw that indeed we were able to reduce the microfluidic speed inside the microfluidic channel and the trapping chamber, that the position of the particle that we were trapping were more centered, further away from the electrode, and we had a stable equilibrium position. We tested it in the lab, and we saw that it was possible to trap and to create large amount of cell aggregates, first with beads, bead clusters, and also that it was working with cell aggregates. But still we had the problem that with only 3 electrodes, we still had cells coming more and more, and that we couldn't in fact stop the number of cells that were constituting the aggregate. So what we decided to do is, still with our design, so this is the PCB that is used to supply both the centering and the trapping, we decided to add a layer of control with LabVIEW with our computer. And the experimental setup is the following. We have a pressure controller from FreeGent, and we are using it to control the flow in our microchannel. We are using first electrode to center the cell directly, and after those 4 electrodes to trap cells. And everything is happening by visualizing the experiment with our MIUI camera, and everything is controlled with LabVIEW. And that's how it looks like. I said we are using those 3 electrodes to perform the trapping. So we are supplying voltage to those 3 electrodes. We are using a pressure controller to continuously supply cells in the microchannel. At some point, what we'll have is a required number of cells. And in this case, we don't want any more cells to come in the main chamber. And we also see that in this case, cells were not forming a dense sphere. So in this case, we reduce the fluid flow, we reduce the DEP force, and at some point, this 4th electrode that I didn't present, we activated. And in this case, it was used, in fact, to seal the DEP cage and the trapping chamber. We decided to let this for 5 minutes, and what we saw is after those 5 minutes, we release the DEP force, we increase the fluid flow, and we saw that right after, in this 50 micrometer channel, cells were staying together. But we are wondering, what about this aggregate? Is it permanent? Can it resist when it's going to a constriction, in this case 40 micrometer in width and 50 micrometer in height? And the answer is here, right here in the video. Cells are staying packed together. They are not reseparating, even though the mechanical stress applied by the microchannel is quite high. And no matter how long we wait, we still see the cell packed together. So it was a relatively fast process to create an aggregate, because we managed to do it only after 5 minutes, to put in perspective with the 10 to 15 minutes used in the system by Sam Yameda. So it's a relatively fast process. So we managed, in fact, to do first to trap cells with a test configuration, then we improved the design to improve the trapping, and then we were able to add a full automation of our microfluidic chip to better control the formation of cell aggregates. Now we have created aggregates, but we said we want to study them. Right now we are deciding the size of the aggregates only by looking at the camera, so we have no real idea of the size of the cell except by looking at the camera or the properties. So what we propose is to use directly an electric field, because we have everything ready on the chip, and to see what is happening when we go from the single cell to the cell aggregate. And that's the following part about the impedance sensing. As I said, the concept of impedance sensing in a microchannel is the following. If you are using a coplanar design, in this case 2 electrodes, pattern at the bottom of a microchannel, what we have is, whenever a particle is passing between those 2 electrodes, we have a current drop. Now, is this design optimal to sense cell? We said maybe not, and we decided to completely study the design and to understand what are the relevant parameters that will influence the signal we'll obtain. What we first decided to vary in this coplanar design with the straight channel is the distance between the two electrodes. What we see is as we increase the distance, we are reducing the current variation, and that's something that is normal because we are increasing the resistivity. So there is no optimal, but we decided to have two different sets of distance between the electrode, one at 50 micrometer, because I said I wanted to have aggregates that would be 50 by 50 by 50, and another set of 150 micrometer, because in this case I could use the liquid electrode concept to kind of have directly an equivalent virtual electrode for our 50 by 50 by 50 aggregate. So once we have those two different distances, we played a bit with the width of the electrode and we saw that there is no optimal. We can always increase the signal, but not so much at some point. So we had to set a specific value of the width, and similarly, we studied, in fact, what would be the angle that we should have in the microchannel, and similarly, we didn't see any optimal. So we had to choose a criteria for choosing which parameter we were going to use. Finally, we decided to have four designs. Two made of conventional design with our straight channel, and two optimized where we tune a bit the parameters. And what we see is for the short design, which means 50 micrometer, we go from the orange curve to the blue curve. So we are increasing the current variation. When we go for our long design with the 150 micrometer spacing, we see that now we go from a quite flat response to a more peak shape response. We decided also to see, because this is happening for a centered particle. Our microchannel is 50 by 50 by 50, let's say, in the zone. We want to see what is happening when the particle is not centered. So we saw that by studying the Y position, meaning the lateral position, that in this case, the variation was quite high for our optimized design. So it means we can't really distinguish between different sizes of particle, and more problematic, we could sense differently similar particles. And it was also valid directly for the Z position. So it means that depending on the height, we might observe different signals, especially for the short design. So we wanted to understand more what was happening, and we performed another set of simulation, and what we saw, in fact, is for the short design, when the particle is very low in the microchannel, what we observe is a double peak, is an M shape. That was valid for both designs. And then we saw a quite significant variation of the current just by playing with the height of the particle. Now if we look at the long design, we observe still an M shape for the long design and the conventional one, but in this case we have a plateau where we can sense all the cells similarly no matter what the height is. And more interestingly, for our long optimized design, we observe a peak, a double peak, but it's not the main one. The main peak is only when the particle is located right here, right in the center of our impedance sensing configuration. And this M shape we looked, and in fact it's not something completely new. For impedance it's not so much seen, but it's very much seen in the case of a coulter counter, which is an aperture in a membrane, and when we apply a certain electric field between those two sides of the membrane, we see that when the particle is going through this aperture and is going quite close to the wall, it's in fact creating a kind of M shape. So based on the result, we went for the fabrication. We fabricated it in the clean room, and then we went for testing. In this case, our experimental setup is the following. We are using a syringe pump to have a constant speed between the different configurations, and we are using an instrument from Zurich Instrument to be able to look at the different impedance design. And what we saw are the curve on top. In fact, it's not so much looking like what we expect, but this M shape that we're seeing is not new. We predicted it in the simulation. It means that in this case, the particles were quite low in the microchannel. So based on those results, we said that we can have different results based on the configuration we want. For the short optimized design, we have high peaks. but we need a vertical centering. For the long optimized design, we have also a peak amplitude that is less sensitive to the vertical position, and we don't really need a vertical centering. And for the conventional design, we can still use them, but in this case, by looking at this M shape and the amplitude, we could get some additional information on the height of the particles. So in our case, we want to be quite insensitive to the height of the particles because we don't want them to influence too much on what we're seeing. So what we decided to go is for the long optimized design. So we decided to take this design and to combine it with our trapping design. And the chip we have is the following. First, we are using those electrodes for focusing cells because we said we need at least to have a lateral centering. Then we are sensing the cells individually, trapping them, and also sensing what we have created afterward, the aggregate. And we have two different designs, one with the auxiliary channels and one without. We are using still the same experimental setup with the pressure controller and the Zurich instrument. And what we saw, that's how it looks in the lab. So we have the microscope, the instrument for the impedance, the control with the computer, and we directly have also the frequency generator. If we look at this video, in this case we didn't focus too much on having a single cell suspension. We will see cell aggregates of different sizes already coming in our microchannel. So we can see that when the first aggregate passes, we have a certain current variation. We are able similarly to trap it, and once we have another kind of cell coming, we still see peaks. And when the size of the small cell cluster is changing, we see different signals. So in this case, by using those 3 electrodes, we're not adding the 4th electrode in this specific case, we saw that it was possible to trap. And then at some point we decided, okay, the aggregate or the simile aggregate we have created is big enough. So what we did is we stopped the DEP force and we re-increased the fluid flow, and what we saw is we were able to sense it directly with the same sensor. So this is just a proof of concept and preliminary result that we can combine both DEP and impedance sensing on the same chip. Now I would like to give you a small conclusion on this work. Let's go back to the requirements we set at the beginning of the presentation. We said we want to have trapping under flow condition. This is done. We want to accept new cells. We can do it in our design. We have a size of the trapping area that should fit the size of the aggregate. This is also performed. We can theoretically combine different cell types. Nothing prevents it directly. We have a stable trapping, and we have a simple fabrication process. However, the cell variability is the first assessment that we did. It should be okay, but we still need to perform further experiment on the cell variability and the effect of the temperature. And last, we are able to sense single cell and cell aggregates. More interestingly, in this PhD, I developed a software, MyDEP, for dielectric modeling of particles and cells. We developed also a fabrication process to have PDMS microfluidic chip and to align them directly on titanium and platinum electrode on glass. We simulated, fabricated, and successfully trapped with our microfluidic chip cells, and we were able to create cell aggregates. We improved afterwards this design, and we were able to perform the full automation of the setup. And last, we also developed a methodology for improving an impedance sensor. As an outlook of this PhD, I would say that there is still some room for work. And for example, we can perform now 3D simulation and thermal simulation to understand better what is happening in our microfluidic chip. We should also perform more viability tests, not in this case just adding tripe and blue, for example in microfluidic chip, but taking back the cells and the aggregates that have been created and put them into culture and see what are their behavior. This is not right now possible with our chip because everything is done to be performed only on chip, not to be taken after. I would also say that we can go back to the initial project and to add an electroporation stage on our microfluidic chip and to create composite aggregates. And I want to show you what would this 2 configuration would look like. In this case, we will use our design with the focusing part, the sensing part. We would create the aggregates here and we will send the aggregate. And afterwards, we would add an electrophoresis stage where we can perform and test different configuration of the electric field to perform the electrophoresis and to sense afterwards what we have created. First application. Second would be to create cell aggregates. If you look in fact at the design, we have different inlets. We have 3 different inlets. So we could use different cell types with different inlets. Let's say type A at inlet 1 and type B at inlet 2. In this case, we would be able to center them, to sense them and to see the different properties, and to create an aggregate of a controlled composition where we know how many cells of type A and type B would be together. This is theoretically possible, however, it still needs some more optimization to do it right in the lab. I would like now to thank you all for your attention, to thank all the affiliation of my 2 labs, and also to thank all my sponsors for this PhD. Thank you very much for your attention.
So thank you very much, Jonathan, for this very clear and professional presentation. So now we are moving to the questions. Maybe we are going to raise. Let's get that.
Can I start? Just a few seconds. Okay. Yeah.
So we start with the floor. The first one is Professor Albert from the Institute.
Okay. Okay, dear candidate, thank you very much for indeed very professional and exact presentation. It showed the breadth of your work. I mean, it's really containing a lot of different elements, going from simulations to development of software, measurements, fabrication, and the whole system integration. First question to you might be, you have done so many things, what element are you most proud of? And this is inspired by my neighbor, in fact.
I would like to mention, let's say, 3 elements. First, the fabrication system, because it can be translated to any kind of fabrication. It's not specific for our design. And it has been translated both in the lab in Lyon, because it was originally developed at EPFL, and also at CEA in Grenoble. So it's something that is not specific to our clean room. We can really fabricate this and use this fabrication process in different parts of the world, no matter the clean room. That's the first thing. The second also is the software, because that's something that can be used by people that are really familiar with DEP, and also people who are not familiar. You don't need a deep understanding on how DEP works to use the software and to see if it's interesting to work with people who have the technology. And the third part is, in fact, what was the core of the PhD is the development of a configuration to be able to trap cells. We knew it was possible to use DEP for creating cell aggregates, but we were not sure it would be possible to trap it and to do it in flow conditions. And that's something that I'm proud of, because it was something that was not completely possible to predict, and it was a bit a challenge, and we were successful doing it, but we couldn't be sure before going to the lab.
Okay, so you're proud of 3 elements. Let's go through these 3 elements, and starting with the fabrication, as you mentioned it. I was also very much impressed by the images that you showed in your thesis, because they really look like very, very sophisticated engineering. You mentioned a couple of in your text a couple of problems that you could encounter, notably the shrinking of the PDMS, and here in your presentation you focused on the how do you say, coplanar surfaces that you could have. Which of these 2 is the most important problem that you overcome with this new technique? And the second question is, what actually goes wrong if you don't do it? Because maybe you can explain that.
So I wrote it in the thesis, but you are right, I didn't mention it when I was presenting. You have a shrinkage problem. Like in our case, at 80 degrees, curing the PDMS or polymer for 2 hours, we have a shrinkage of roughly 1.5%.
Yes.
And that's something, in fact, that is not so much visible if you work in a very small area. But if you want to work on a larger area, like in our case, we are working on, let's say, 3 millimeters from the one side of the chip to the other side, this effect is directly visible. So you can't align it on all the structure. So that's something you really need to be careful about. But this shrinkage is also linked to the PDMS you have. What is the thickness of the PDMS and your curing condition? If you modify the temperature at which you are performing the curing, you will change the shrinkage. Similarly, if you change the thickness, the shrinkage will change. So those are important elements that will influence and kind of prevent you from performing the alignment, even if the chip is completely flat. Let's say the flatness is more an operational requirement. If you don't have the flatness, you either need to be very skilled with your fingers, or if you want to use a standard, let's say, machine like the one I use in the cleanroom, yes, you need to adapt it, but you can work with it no matter what is the thickness. You just need to work with the flatness.
Okay, so you overcome the shrinkage by changing temperature, did I correctly understand that?
No, in fact, to overcome the shrinkage, it's 1.5%. Either I could work at room temperature, because in this case, yes, you don't have any shrinkage, yeah, but the problem is that the created PDMS doesn't behave like the PDMS we are used to. So it's extremely sticky, and the problem is that it might not be completely cured. And if we expose it to any higher temperature at some point, you will observe an additional shrinkage that is not predicted. So it's more difficult, it's less predictable structure.
So this is not completely solved, in fact? Or?
In fact, for the shrinkage, what I did is, in fact, I increased the design of 1.5%, the size, meaning that, in fact, when I have the shrinkage, I am able to compensate back this problem.
Okay. So there's a practical solution, in fact.
Yes.
Okay. Very good. The second element you mentioned is the software, the MyDAP, I think. And indeed, it's a very impressive piece of work, and I think I agree with you that I guess many people will profit from this software. Nevertheless, I have a couple of questions about this software, and let me just go through a bit through this chapter. Let me see. You have a couple of cell models, in this case page 76, and you also showed it in one of your slides, figure 2.3, where you have homogeneous particle, single cell, 2 shell, 3 shell. Oh, it's already there. Yeah, very fast. And a 4 shell also implemented, you mentioned.
Yes.
It's very impressive. The formulas become more and more complicated, but my question is, how realistic is this representing real cells? Because in real cells you have a lot of subcellular compartments with all individual envelopes in their individual, and they are not all at all, I would say, concentric.
Right.
So how realistic is this, and how can you incorporate a little more of the realistic cell?
So at some point we had to decide what we would model. And if we want to model, let's say, vesicles inside the cell, the problem would be the position. And if the vesicles are located only in some side of the cell, it would be way more difficult to model it. So in fact, we decided that it could at least be approximated by the 3 shell model. And by playing with the size, we could at least have a kind of equivalent vesicle that would be either the nucleus or the equivalent vesicle that could be modeled. So in fact, just to elaborate on this answer, there is the 4 shell model. And some may say, why the 4 shell model? Why are you not stopping with the 3 shell model? And the answer I got from the person who published this cell model, Ralph Olsen, and he told me that in fact this is the only fitting that was giving good result for his for this case. He couldn't do it with 3 shell, with 2 shell or single shell. He had to go for the 4 layer, which, and if we look through the article, makes sense in case of what layers are important and how big the change in the properties impact the response.
Okay, but you agree with me that certain cells do have asymmetric envelopes inside. So can you give an estimation about the error that you would make? Because I can understand you cannot completely model it, but if you at least can say, well, the error that we make is less than 2% or 5% or something, is that possible to compare that?
Right now, this is not possible. It's not something I can estimate by hand or that we have developed the software for.
Okay.
It's more for a general understanding. It's not really, I mean, the software is not ready, let's say, to go back to a fitting, because this is more or less what you were asking.
Yes.
Could we go back and, by analyzing the signal, go back to what is the original model?
Yeah.
And to
no, I think we also discussed that in the past a couple of times, and maybe not completely for fitting, but for the sake of reality check, I wonder if your measurements in, I don't know, figure 2.8 or so, or other 2.6, you always take, I guess, data from literature about yeast cells or HEK cells.
Yes.
or so. But did you ever perform a measurement on these cells to compare it to what you model, and how close is it? I mean, not that you have to fit it, but to get an idea how good it is.
So the answer is no, no direct measurement. And also, elaborating on this question, for using some, let's say, a yeast cell model, we found many models that were published in the literature.
Yeah.
And if we put all of them together, because some are two shell, single shell, full shell, there is no fitting between the different parameters. So it means that we have a discrepancy in the curves. So let's say the software is not done to say, this is the right model. It's just to provide what has been published and to say it has been accepted by the peers.
Yeah.
So this is what we can propose, at least as a starting point.
Okay, well, I agree that, let's say, the model probably is very useful to see trends. If you have a little larger cell or a little different shape, what would be the effect on the impedance or so. But could you give me some idea about the variation that you would expect from a single cell measurement compared to a model or impedance curve that you simulate in your model? Is that really quite close, or can it be really different? Because I can imagine some people using your software and then they measure and they see something probably a little bit different. What would be acceptable for you to say, well, this is more or less okay?
So the question is, how did you get the data?
Well,
because for, let's say, getting those data, they are mostly taken by electrorotation, not by impedance.
Okay.
And if we want to go for impedance, the question is, what is the configuration you want to work with? Is it the one with facing electrode, or is it the one with coplanar electrodes? And depending on the result you have, you need an additional layer of modeling. You need a modeling of what is called the volume fraction.
Yeah.
And the problem is, if we want to work on this theory on the volume fraction, it is, let's say, a mathematical object, and we have to go through something that is linked to conformal mapping. And let's say for the facing electrode, some theories have been developed, mostly by in the laboratory of Iwan Morgan and also in the laboratory of Philip. But when we want to go for coplanar electrode and sensing the single cell, nothing has been published. And that's also the reason why we did this work on the impedance. We wanted to understand how the cell will influence, and just by the fact that the height of the particle in your microchannel for coplanar design might change the impedance signal you are seeing, it's realistic to say that we can't really get the values of the parameters directly from the curve.
But if you look at figure 2.7, you have a simulation for different values of conductivity.
Yes, of the medium.
Of the medium, exactly. But then it also depends on the shape of your structure then, I guess.
Yes, in this case it was done for spherical particles.
So how would this translate to a measurement setup? Also a spherical, theoretical electrodes surrounding the cell. Is that what you would have then, or?
So if I
2.7?
2.7. So 2.7, this is the sweep, in fact. I mean, I don't have it, I think, in the presentation.
No, okay, but I just meant take it because you have different conductivities,
Yes.
from 1.6 to 1 millisiemens to 1.6 Siemens. So it's a big difference.
I will just put, I have the one for the Jurkat cells, just as a
okay, this is okay. So my question is, the model you use here is a spherical model, you say?
Yes.
So suppose in an ideal world you would have a spherical electrode somehow.
Spherical cell.
Yeah, the cell. But this is looking at the CM Clausius-Mosotty factor.
Yes, this is the real part of the Clausius-Mosotty factor.
Yeah, in the combination of the cell with the medium.
It's the real part of the Clausius-Mosotty factor of a cell in suspension in a medium, but there is no here linked with the configuration of the electrode.
Okay, well, I'm just asking these questions because suppose at some point a person is measuring a single cell in a very sophisticated small setup enclosing electrodes enclosing the cell. How can he or she use your simulations here? It's just to find the optimum CM factors, let's say, where at what frequency is the highest contrast.
Yes, she could use it for the higher contrast to understand when the force should be higher.
Yes.
Let's say in this case this is the real part which is linked more to dielectrophoresis, but if she's performing, for example, electrorotation, it will be more linked to the imaginary part, and this is something that can be checked. If the speed of the particle is maximum at this frequency, so it could be used. As I said, the fitting is not possible in the software, but you can also plot your data and also check manually with different values what is the result.
Okay, clear. I would like to move on to the trapping design and simulations and results that you have obtained. Again, I think it's a very original and sophisticated approach with the 8 electrodes at the trapping chamber. And I would like to discuss with you the figures. I don't think you have them in presentation, but in page 102 and 103, figure 3.6 and 3.7. You probably know them by heart. But it shows very nicely in a three-dimensional way plots of, I guess, energy or forces.
Yes.
And maybe you can explain, well, my interpretation between 3.6 and 3.7 is 3.7 has a dimple. That is a stability point for the cells. Is that correct? Or?
So figure 3.6, it corresponds to let's say more elaborated figures of the deep force, and in this case this is only the longitudinal deep force, so meaning along this axis. So we see that we have one trapping position, and the other figure 3.7, is the same thing, the same force balance, but in this case in the y direction, to understand if there is really a trapping position or if there are trapping positions.
But okay, if I look at the figure E in the first one, I see X, Y, Z. It's oriented in the same way as in figure 3.7.
This is the geometrical orientation, and it's a free. Let's say it's
so it's a different X and Y, in fact, here.
No, it's the same, but let's say the Z coordinate is trying to find an illustration of the representation of those two forces, to try to see when those two forces intersect, meaning the drag force coming from the fluid and the dp force coming from the electric field. This is more a representation of what force predominates in which area. That's why you have the, let's say, a side view on figure E and more a top view, which is the one we want to look at, in figure F. And in this case, you see that you have a part that is green, which means that the drag force is predominant, and you will bring the cell towards the outlet. And after a specific line, it's figure F, you would see that the cell would be trapped. And what I elaborated in figure 3.7 is the same, but in this case, if you add the component coming from the Y, the particle will get, in this case, center laterally. So you mean, if I represent it with the figure, the particle will come in your microfluidic design, it will get trapped, and if it's already in the center, it will stay here. But if the particle is coming from the side, in this case, it will be trapped at a higher position, or not higher, but let's say more towards this electrode, and then, because of the Y force,
it will move to the middle.
Be center.
Yeah. Yeah, that's also what I sort of interpreted, but I thought that in this figure F, it could sort of move along this dotted line while still maintaining on the same line, and the next one it will go to position X, let's say, in the second one.
So in fact the problem is that these DEP force depend on the axis. It's along one, the X component, Y component.
Okay.
So let's say the figure F is the X component,
yes,
and after I look at the Y component,
and that has
to decouple, because it's not really possible.
No,
to
you cannot do it in you need 4 dimensions to show it,
yes.
Something like that. Okay, excellent. I think I understand that more or less correctly then. And then we move to the, what is it, the aggregate formation. And, oh yeah, then I come back to a very basic question, which I didn't ask you yet. The cell aggregates, you mentioned in the beginning it's sort of between a single cell and a tissue, or maybe even piece of organ, or functional piece of organ, or something like that. Is there any physiological relevance to looking at aggregates? I mean, I understand you want to do more than one cell, but does it happen in real life, aggregates? And where? In a organism?
So you have the formation of erythrocyte aggregates in the body at some point. So this is one.
In blood.
Sorry?
In blood.
In blood, yeah.
No, that was also my point, that if you have blood clotting, that starts with aggregation, but you don't mention anywhere in the text, so I thought it could be some...
I think it's mentioned once.
Oh, okay, I missed that, sorry. Because it's a very important aspect, thrombus formation.
Yes.
It's very relevant for any kind of diseases, and maybe this technique allows you to study it and maybe even to resolve it or prevent it. I don't know, but I mean,
at least to try to understand.
Yeah, exactly. So
that's one application. So this, let's say, for the relevance of this work, you're right. It could be applied to this kind of physical concept.
Yeah.
But also, if we want to work towards the aggregate, we can't really directly work with an organ on chip. It's not so easy to create it and to study it. So at least what we are proposing with the cell aggregates, it's, let's say, a more realistic model than the single cell, because at least the cell in this case have a surrounding. It has neighboring cells, and it can have a contact with the other cells. I completely agree, there is no vascularization, there are many limitations, but at least the cell can be in an environment that is a bit closer to the tissue than when it is in suspension alone.
But what you miss is cell-cell interaction, and I mention it because you show that once they are sort of aggregated, they stick together. So they already form a sort of membrane contact, and that already gives a different situation than having them just freely close together.
Yeah, so that's when I said they have an environment, meaning like they don't have a cell that is far, they have a cell that is close to it. So you have one cell, another cell that is getting into contact, and we also have, we think, an effect of we have two dipoles, in fact, that once they join each other, you increase the interaction because of the dipolar effect.
Yeah.
So you in fact put, let's say, the cell more into contact thanks to DEP than if you just put them into contact without using an additional force.
But you could get a sort of membrane fusion then, or not?
It could be like cell fusion, but...
yeah, or not complete fusion, but just the membranes could
at least the membranes are getting very close to each other, and we postulate that you might have cadherin-cadherin interaction between the different membranes.
Can I continue still, Mrs. Chairman? Yes, okay. I would like to go to your last part, which is the impedance sensing of the single-cell aggregates. Because I have a couple of questions there. You say that you have the coplanar and the, how do you call the other one? Well, the opposite electrode design.
So facing electrodes.
Facing electrodes, where the facing in principle is a more homogeneous field, which is better, position independent or height independent, but you say it's a more complex fabrication process.
Yes, because you need to perform an alignment between two different layers. And while this alignment in this case, in my case of the fabrication, was quite possible, because you have the bottom where you have the electrode and you are bringing a surface where at some point the two will be in contact. So you don't need to change so much the focus of the machine you are using. The problem is with, let's say, those facing electrode, you need to change the focus all the time to perform the alignment. So this is way more challenging. And in fact, if you need a very precise alignment, you start to have some limitation of the machine to perform those chips with top and bottom electrode, which I call the facing electrodes.
It's a sort of practical consideration.
Yeah, mostly practical consideration.
Okay. I also wanted to ask you, is it necessary to have contacts also at both sides, you think? Or to have electrical contacts, because that's also sometimes limitation of the technology.
So in this case, in my case, the thing was to be able to use the chip directly with as little processing as possible.
Yeah.
And in this case, I can just use the two sides. If I have everything on the same side, sometime I may have to, in fact, insulate the electrode to have them to go under the microchannel. So that was more, in this case, a practical consideration to choose to have the two sides. Plus, we also already had the technology in the lab, so which means.
Yeah. Okay, and then you work. You do a lot of simulations on, let's say, the channel and the constriction and widening of the channel. And I think I get the idea that you, let's say, want to optimize the signal coming from the cell when it passes through a narrow opening, so that it has maximum effect, while it's not directly in the area of the electrodes or something like that. Huh? What I see is that you have a better, let's say, independence of the electrode height, or the height of the cell, but the signal is going down in strength. Huh? Yeah. This is it.
In this case, we are looking at the relative current variation.
Yeah, yeah,
so
but
we are
also
not directly looking at the signal, because we are normalizing the signal in this case. This is in percentage. This is not in.
Okay, it's only variation of the signal, so it's less.
Not the absolute value. You have one figure in the thesis, which is in the supplementary material, where you have the absolute value.
Okay, so the variation is getting better, less variation. As a function of the height.
In this case, it depends. The reason why we decided to go for the relative current variation is to be able to compare similar things.
Yeah, but you also need to look at the signal strengths, because if you have 0.001,
exactly,
you don't see it.
And that's also why it's important the next part with the fabrication. The test, because we saw that at least we could detect it, and we obtained similar results.
Yeah.
And we saw that at least the signal-to-noise ratio was sufficient enough for us to get some signal.
Yeah. Okay, then maybe the last remark I want to make here is that at the very, very last part of your even conclusions or recommendations or outlook, I see all of a sudden in page 159, section 7.2.5, I see a 4-electrode configuration. I think it's the first time I read it there. And I was thinking of it earlier. Can you improve your measurement signal or whatever by using 4 electrode signals?
The 4 electrode is 3 for trapping and the 4th one for sealing the microchamber. So in fact, we have 3 active electrodes that was used for the trapping, and that's something I showed earlier.
Oh, this is for the trapping. But how about the sensing, the impedance sensing? Can't you just impose a current and then measure with 2 very small electrodes, very closely spaced, high impedance, measure the voltage, and then you're completely independent of everything? Only when the cell passes between 2 measurement electrodes, you see a difference in voltage drop over there.
I'm just afraid that the signal will be too small. If we have it needs, at least for our design, it would need, for example, if you have, it's a 4-point measurement you are speaking about. It means that you would have electrodes that are even further away.
Yeah, but yeah.
And at this point, I'm afraid that.
But you impose a current, and it's really totally independent of how big your electrode is. You just push in the current, and you know the current is going, and then the two measurement electrodes can be very closely spaced, so that they only measure when the cell is in between, they see a difference.
It's true, but it means that in this case, if we start to use more electrodes, we have a bigger volume. So it means that if, imagine we have 2 additional electrodes, it means that if a cell is in between the different electrodes, we would see it differently than when it's at the center of the trapping, of the sensing unit. Like all the work that was performed in this case, the maximum is here. And the problem is, it's nice if you have one cell, but if you have one cell here and another one that is just before, in this configuration it's working. But if you have more electrode, you increase the risk that you have many cells at the same time in the sensing unit.
Okay. I am satisfied with this answer and all the other answers as well, so I would like to give the word back to the president or the other opponent.
Thank you very much. So now we move to the second contributor, who is
Merci Madame la Présidente. Bon, pour l'audience, je vais poser mes questions en français pour faire une alternance avec l'anglais. Donc tout d'abord, je vais te remercier pour ta présentation qui montre une rigueur dans la démarche que tu mènes autour de tes travaux de recherche. C'est toujours carré, c'est net, c'est très très propre. C'est un peu comme le PDMS, c'est sans bavure, voilà, c'est très très propre. Mais bon, avant de poser des questions, je vais remercier tes encadrants de m'avoir sollicité pour participer à cette audition et pouvoir étudier tes travaux un peu plus en profondeur. Donc je remercie Marie, Philippe et François de m'avoir invité. Merci à vous, c'est pas la première fois que je viens, j'espère que ça continuera. Je viens toujours avec plaisir. Et puis je pense que pour toi c'était pas forcément facile, pour une simple raison, c'est que quand on voit le contexte de ton travail, il y a un poids historique qui est assez fort, puisque le laboratoire Ampère a maintenant une reconnaissance dans le domaine de l'interaction cellule-champ, avec les travaux que tu as présentés, les travaux de Marie. Donc c'est vrai que quand on arrive dans un laboratoire qui a cette expérience et qu'on te demande de travailler sur cette interaction cellule-champ, c'est pas forcément évident, parce qu'il faut s'approprier les travaux de tes encadrants. Et puis tu avais aussi une interaction forte avec Philippe Renaud, qu'on ne présente plus. Et donc il fallait pouvoir s'insérer dans ce contexte historique, et je trouve que tu l'as très bien fait. Et c'est aussi pour une grande qualité que tu dois avoir, de pouvoir t'approprier ce qui existe dans un laboratoire pour arriver à ce qu'on voit ici, un lab on chip qui est capable de faire tout ce que l'on rêve de faire en interaction champ électrique-cellule. On a une formation, je pense, à peu près similaire sur le génie électrique. Donc on est très sensible au champ électromagnétique. Et l'application vers le bio, c'est quelque chose de fantastique pour nous qui venons du génie électrique. Moi, je sais pas comment on dit ça, c'est super, quoi. Voilà. Donc c'était ce que je voulais tout d'abord citer. Et puis ton jury, il est aussi assez notoire. Tu as quand même tous les spécialistes, soit de la thermie, soit de la dielectrophoresis, ou de l'intégration de ce type d'électrode sur puce. Donc c'est aussi bravo à tes encadrants d'avoir constitué le jury. Voilà, je me sens tout petit en fait dans ce jury, mais bon. Voilà, et puis il y a aussi la microfluidique avec Anne-Marie. Voilà, donc je vais arrêter ici pour ces remarques sur le contexte de ta thèse. Tu as cité les 3 points qui étaient clés, je les avais notés, c'était MyDep, la techno et puis le design. C'est vrai que c'est les points marquants de ton manuscrit, surtout le logiciel. Alors moi ça m'embête un petit peu parce qu'avec mes élèves j'ai l'habitude de leur faire sous console toutes les simulations, et là je suis un peu embêté. Maintenant il va falloir que je leur cache qu'il y a MyDep. Ça c'est pas sympa.
Si c'est avec console c'est bon, parce que c'est pas intégré avec la simulation des électrodes.
Oui, mais bon, on les connaît, les étudiants, s'ils trouvent la réponse, ils vont pas aller la chercher tout seuls. Voilà. Alors là où j'étais un petit peu frustré, c'était sur les aspects bio, où effectivement c'est une thèse qui se veut, alors peut-être que j'ai extrapolé un peu trop, mais qui se veut à l'interface. Et c'est un peu les points qui m'ont un petit peu manqué, c'est le lien direct avec la biologie et les aspects physiologiques qui nous permettent d'élaborer tous ces modèles et de manipuler le vivant à l'aide du champ électrique. Voilà. Pour nous qui sommes du domaine, c'est assez simple de comprendre tout ce que tu présentes, c'est de la physique finalement, tu fais de la physique. Mais je me pose la question, si tu présentes MyDep à un biologiste, comment il fait?
Je dirais que c'est déjà plus facile pour un biologiste d'utiliser MyDep que de sortir MATLAB et puis de commencer à tout reprogrammer. Donc c'est déjà un premier élément de réponse. Le deuxième élément de réponse, c'est la base de données. On a une base de données qui compile plus de 80 éléments de la littérature, ce qui permet à un biologiste de ne pas commencer à refeuilleter toute la littérature, mais de dire, ben voilà, on a différents types de cellules, et il n'y a pas qu'un seul type, qu'un seul modèle, il y a plusieurs types de modèles, et finalement, peu importe le modèle, le biologiste, ce qu'il va regarder, c'est, on a tel modèle, on a finalement tel, imaginons des cellules des HEK, il va regarder, il va dire, voilà, c'est tel modèle, ok, c'est un single shell, mais ce sera un two shell. On va dire pour l'utilisateur lambda qui n'est pas spécialisé à dielectrophoresis, ça ne va rien changer. Donc disons que ça va permettre une approche qui est beaucoup plus aisée pour un non spécialiste que de commencer à tout recoder sur MATLAB. C'est le premier élément de réponse. Le deuxième élément de réponse, c'est que utiliser finalement ce logiciel, pour l'avoir fait avec des biologistes, ça va donner envie d'essayer. Disons que ça donne envie de dire, bah voilà, mais est-ce que ce serait possible de travailler avec des bactéries? En fait, tous les développements qui ont été faits sur les ellipsoïdes, ils ne viennent pas de nulle part. C'est parce qu'on a discuté avec des biologistes et ils ont dit, ah ouais, mais est-ce qu'on pourrait travailler avec des bactéries? Et du coup, de là, toute l'étude de littérature et de comprendre comment implémenter ça dans le logiciel.
Oui, et à l'inverse aussi, pour les physiciens, c'est pas forcément évident de trouver le lien avec la biologie. Voilà, donc tu parles de bactéries, c'est vrai qu'il y a la paroi, il y a la membrane, et voilà, je pense que une évolution de MyDep, c'est peut-être avoir ce module pédagogique, ou je sais pas, une partie, un tuto.
Alors,
toi, qui explique un MOOC, ouais.
Effectivement, alors pour l'instant...
Parce qu'on parle de modèles simple COP, double COP, triple COP, X1000 COP, mais si on ne fait pas le lien avec la constitution intrinsèque des composés qu'on manipule, bon, aussi bien le physicien que le biologiste, ils ne vont pas réussir à travailler ensemble. Donc un de nos rôles, c'est d'arriver à faire ça.
Je suis complètement d'accord avec ce point. Là, on s'est plutôt focalisé sur...
Il y avait un autre point, ce qui m'a un petit peu manqué, c'était ces modèles-là, le simple coq, il suffit si on est en fait en très basse fréquence. Et le lien avec les gammes de fréquences qu'on peut utiliser, est-ce que ça peut influencer le choix des modèles?
Alors effectivement, si on travaille avec la très basse fréquence, bon, on ne va quasiment pas pénétrer dans la cellule, on va principalement regarder les propriétés au mieux de la membrane, quand on va commencer à monter en fréquence, donc c'est un modèle qui est satisfaisant. Donc je suis complètement d'accord que la gamme de fréquences dans laquelle on va travailler, plus on va vouloir monter en fréquence, plus il faut qu'on ait un modèle qui soit fin, qui soit capable de modéliser. Pour revenir au point précédent, donc le travail de MyDep, c'est un travail que j'ai réalisé dans les 4 premiers mois de mon doctorat en MATLAB. Et toute la difficulté, ça a été de translater ça pour arriver à publier quelque chose, à le rendre accessible. Et la difficulté de faire quelque chose en MATLAB, c'est que ça fonctionne sur sa machine, mais dès qu'on change de machine, dès qu'on change de version de MATLAB, dès qu'on change de système d'exploitation, plus rien ne fonctionne. D'autant plus que MATLAB met à jour tous les 6 mois la version et que ça change complètement le fonctionnement de l'interface. Donc en fait, c'est toute cette difficulté à rendre reproductible le travail scientifique qu'on avait réalisé par le biais de l'interface. Et c'est aussi toute l'importance du travail collaboration avec Olivier. On a passé tout ce qu'on avait développé tout ce que j'avais développé seul en MATLAB, qu'on a transformé en Java, pour avoir quelque chose qui est multiplateforme, qui dure dans le temps, et qui nous permet de savoir que dans plusieurs années, si quelqu'un utilise ce logiciel, il suffit qu'il ait Java pour pouvoir l'utiliser.
C'est un apport indéniable, ton travail sur MyDep. C'est un des gros points forts, je pense, de ton résultat. Moi, je trouve ça super.
Et donc, pour continuer, ce que je voulais dire, c'est que ça a pris 3 ans pour arriver à publier ce travail. Donc, on a principalement mis l'accent actuellement sur le logiciel et l'utilisation du logiciel. Donc, on n'a pas un tutoriel pour dire quel type de modèle il faut utiliser, mais on a un tutoriel complet sur le site web, sur notre page GitHub, qui explique toutes les fonctionnalités de l'interface et comment on les utilise. Donc là, la prochaine étape, c'est de faire des tutoriels et puis de dire voilà, si on veut faire ça, quelle est la validité, pourquoi est-ce qu'il faut faire ça, il faudrait le faire comme ça.
Très bien. Alors ce qui est aussi très intéressant dans ton travail, c'est tu es parti sur une structure à 8 électrodes. Quand j'ai découvert ton manuscrit, j'ai dit mais qu'est-ce qui s'embête à faire? Et en fait c'est super pratique parce que c'est versatile. C'est, je pense, c'est ce qui t'a permis d'aller à cette structure simplifiée qui permet d'optimiser ce qu'on veut faire. Mais je pensais qu'avec cette structure à 8 électrodes, vous aviez d'autres idées derrière la tête. Est-ce que tu peux en dire plus?
Alors, donc là je vais remettre la structure à 8 électrodes. Donc on va dire cette structure à 8 électrodes, l'idée de base était la suivante. Je vous ai montré les travaux de Marjane préalablement où on avait 4 électrodes. Donc on va dire la 1 et la 5 et la 3 et la 7. Et ces électrodes étaient utilisées pour faire du sensing, pour regarder l'anisotropie. Donc c'est-à-dire qu'on a une fréquence entre deux électrodes qui sont face à face et une fréquence entre deux autres électrodes. Et l'idée de base de mon travail, c'était de me dire, finalement, si je reprends cette configuration et que j'ajoute du piégeage in situ, en rajoutant ces électrodes, est-ce que j'arriverai pas à combiner la dielectrophorèse et l'impédancemétrie? Et donc c'est comme ça que je suis arrivé d'une configuration où on était en sensing à une configuration où cette fois on allait avoir des électrodes avec différentes forces qui seraient là. Donc les électrodes 4 et 8 et 2 et 6. Sauf que ça, c'est valable si la cellule est déjà arrivée. Si on veut arriver à piéger en flux, et ça c'était toute la complexité de ce travail, il faut arriver à trouver quelle est la meilleure configuration. Pour être honnête, la configuration de départ avec ces 8 électrodes, ce n'était pas la configuration à 3 électrodes, c'était une configuration avec 5 électrodes. Le problème, c'est qu'au fur et à mesure des réflexions et des différentes courbes qu'on avait, on a vu qu'on n'avait pas une linéarité, grosso modo, du champ, et on s'est dit peut-être qu'on peut faire mieux. Et c'est là tout l'intérêt de se dire on va faire une étude systématique pour essayer de comprendre quelle est la meilleure configuration qui permet de piéger. Et donc ça, c'est un petit peu le pourquoi des 8 électrodes, et puis après, finalement, faire face à la réalité et de dire en fait ces 8 électrodes, c'est très versatile en laboratoire, mais quelle est la meilleure configuration qui nous permet de piéger les cellules en flux? Et ça, c'était un travail en simulation, mais surtout un travail de test en laboratoire, où finalement on a testé plusieurs configurations, pas uniquement celle à 3 électrodes, ce que j'ai détaillé un peu plus dans le manuscrit, mais pas forcément au niveau de la présentation.
D'accord. Alors, sur... je pense qu'il y a une question, tu sais, que je vais te la poser. J'aurais pu commencer à l'inverse. Alors, première question, quelle question je vais te poser? Donc tu en as parlé sur, à un moment donné, tu pièges des cellules et puis on voit le bleu de trypan qui s'échappe. Et alors, pourquoi? Quelle est la raison profonde qui fait que ce bleu de trypan s'échappe?
Qui pénètre dans les cellules.
Oui, enfin...
Non. Là, les hypothèses qu'on a, ça peut être un effet lié à la température. En fait, on va exposer la cellule à une température trop forte. Donc là, elle va commencer à pas vraiment apprécier. C'est une première chose.
Justement, dans tout ton exposé, on ne voit jamais la valeur du champ électrique. Pourquoi? Est-ce que c'est une volonté ou...
Non, c'est pas une volonté. J'ai préféré, comme je discutais du piégeage en lui-même, présenter le gradient du champ au carré au fur et à mesure et non pas la valeur absolue, non pas la valeur du champ. Donc la valeur est présente dans le manuscrit et on voit qu'on est légèrement au-dessus des seuils d'électroporation. Il n'y a pas non plus 1 ou 2 ordres de grandeur de différence.
Est-ce que c'est quand même un peu embêtant alors? Non.
Ne pas l'avoir mis dans la présentation?
Non, de justement le bilan que tu as fait, il n'y a que des traits verts, puis il y a un trait rouge.
Orange.
Orange, c'est vrai, donc ça veut dire qu'on peut encore passer. Voilà, ce petit point orange, est-ce que comment s'en affranchir, parce qu'il est relié au champ électrique?
En fait, il va être relié au champ électrique et puis aux considérations thermiques. Donc la question, et en fait, c'est tout le travail de la configuration, c'est d'essayer de faire en sorte de réduire au maximum la tension de piégeage pour arriver à diminuer les éventuelles influences du champ électrique. Et ça, je pense que c'est un travail qu'on a amorcé, mais qui pourrait être encore amélioré. Là, après, il faut trouver d'autres idées pour avancer.
Puisqu'on est sur ce point-là, est-ce que je peux vous poser une question quand vous avez la posée?
Bien sûr, Madame la Présidente.
Justement, dans les effets thermiques, est-ce que vous avez évalué les effets thermiques? Est-ce que vous savez de combien s'élève la température localement ou globalement? Est-ce que c'est quelque chose que vous avez un petit peu
on n'a pas étudié, on a fait quelques simulations, mais c'est un travail en cours. On n'a pas des résultats probants à exposer. Et c'est quelque chose qui expérimentalement qui est assez difficile à quantifier. C'est pas quelque chose qu'on peut regarder et avoir une information comme ça. Notamment, c'est ce que j'expliquais aussi au niveau de la conclusion et de la outlook, la difficulté de quantifier cette température va venir du fait que par un microscope, on va être sur le plan de focusing qui serait tout en haut du PDMS par exemple ou tout en bas, mais on va être difficilement dans la chambre.
Il y a quand même des modèles qui permettent d'estimer la quantité d'énergie qui est injectée et d'en sortir une élévation de température maximale.
C'est vrai, mais ce genre de modèle dépend beaucoup aussi de la configuration dans laquelle on est, avec comment sont placées les électrodes, sachant que le modèle qui est publié, et je pense qu'Anthony Ramos voudra en discuter tout à l'heure, c'est pour des électrodes interdigitées, donc c'est pas forcément pour la même configuration.
Mais ce qui compte, c'est l'intensité du champ au final.
C'est un sigma avec carré,
ouais, sigma E2.
Sigma E2 avec le K pour on va dire la capacité thermique, pardon, la conductivité thermique qui va importer. Donc c'est aussi pour ça qu'on n'a pas fait de calcul exprès en simulation, puisqu'on s'est dit qu'on allait regarder ce qui se passait expérimentalement, plutôt que de faire des modèles très complexes et de penser que tout allait bien et puis après d'aller dans le laboratoire. On a préparé tout le reste et on a voulu voir ce qui se passait en laboratoire et après de regarder en simulation si effectivement ça fonctionnait ou pas. Et qu'est-ce qu'on arrivait à avoir. Et ça, c'est un travail qui est en cours, la quantification de l'élévation de température à l'intérieur de la chambre.
Parce que, moi, quand j'ai revu tu peux nous remontrer la vidéo de trapping des cellules pour l'élaboration du... Alors, je ne sais pas si on doit dire organoïde ou sphéroïde. L'agrégat, voilà, où on voit qu'il se met un petit peu à tourner, on voit les cellules qui s'accumulent et puis ça tourne un petit peu, les cellules tournent entre elles. Alors la raison de cette rotation, elle est liée à l'écoulement ou à un petit effet thermique?
Alors, pour l'effet thermique, c'est quand même assez loin. Par contre, pour la rotation, on a quand même du fluide qui arrive en permanence au niveau de nos cellules.
J'ai pas compris pourquoi tu dis que l'effet thermique
là, on est
est assez
quand même
loin.
Relativement loin des électrodes et a priori, c'est quelque chose qu'on n'a pas observé quand on a élargi la chambre. Cette rotation des cellules, elle ne tourne pas sur elle-même. Donc ça, c'est un premier élément. Et la deuxième chose, c'est que les cellules commencent à tourner une fois qu'on en a deux. Donc une fois que le fluide commence à venir interagir avec plusieurs cellules. Donc c'est des éléments qui nous incitent à penser que c'est principalement un effet dû au fluide. Mais c'est sûr, on n'a pas la certitude que ce ne sont pas aussi des effets thermiques qui viendraient influencer tout ça.
D'accord. Et alors, après, tu nous as montré la vidéo qui montre que les cellules sont plutôt... ça, c'est une question qui était plutôt assemblée. Hein, c'est-à-dire en 5 minutes, tu dis qu'il y a un début de cohésion. Alors, c'est vrai qu'en général, on parle de protéines qui font qu'il y a des liens qui se créent. Hein, mais potentiellement aussi, tu peux avoir de la perméabilisation qui est liée à un effet du champ, qui fait que l'agrégat commence à apparaître et il y a une fusion entre... Enfin, il y a des adhésions qui se font aussi par effet thermique. Est-ce que c'est des choses sur lesquelles tu peux faire un commentaire? Alors, ce n'est pas des choses qu'on a quantifiées. On a fait les mêmes tests aussi avec du trip and blow et ce qu'on a vu, c'est qu'on ne voyait pas de trip and blow à l'intérieur des cellules quand on l'utilisait. Mais ce n'est pas des choses dans lesquelles on est rentré plus en détail. C'est aussi pour ça que je n'ai pas souhaité le présenter à ce moment-là. D'accord. Alors, un de tes enjeux de début de thèse, c'était de faire un procédé simple. Alors, le résultat que tu obtiens, moi, je le trouve fantastique. Pour les effets d'alignement, c'est vrai, quand on voit que tu es sur une longueur qui fait 2 cm, un petit peu moins, 1 cm entrée à sortie. Tu annonces un désalignement inférieur au micron, sachant que c'est 2 objets qu'on doit activer. On doit activer la surface et puis les mettre en contact. C'est vrai que c'est un très beau résultat. Donc en plus, tu le diffuses dans les autres laboratoires, donc ça, c'est super. Comment tu positionnerais cette solution par rapport à une géométrie fluidique élaborée avec de la SU8 que l'on aurait photolithographiée et ensuite packagée? Alors la question, il y a plusieurs éléments de réponse. La question, c'est comment on va packager la SU8. Généralement, ce qui est fait, c'est fait avec des choses qui sont réversibles, où on va venir appliquer un couvercle sur la SU8. Le problème, c'est qu'on a souvent l'enjeu que si le couvercle n'est pas serré assez, on va avoir des fuites, et si le couvercle est serré trop fort, on peut avoir des problèmes au sein de la SU8. La deuxième réponse, c'est la SU8 en elle-même.
c'est quelque chose d'assez capricieux souvent. Et dès qu'on va avoir une modification des conditions de fabrication, une modification de l'humidité notamment, ça va introduire des défauts. L'autre élément, c'est que la configuration qu'on fabrique en SU8 va influencer le design même du canal microfluidique, va influencer finalement la réponse de notre SU8. On va avoir des craquelures qu'on peut arriver à résorber, mais il faut faire des considérations supérieures au niveau du design sur quels doivent être les endroits pour relâcher les contraintes au moment même du design. Et donc je dirais que c'est possible de faire des choses similaires en SU8, mais la question est après comment on vient fermer la chambre microfluidique.
Voilà, comme ça sera de façon plus interactive, et puis comme c'est dans le même paquet de questions, ça évitera de revenir. Il y a maintenant des technologies à base de films secs.
L'ordile par exemple.
Alors ordile, il y en a plein d'autres, hein, qui permettent justement de capoter sans avoir recours à de la compression, des choses de ce type-là. Donc ça, ce sont des technos qui sont matures. Alors effectivement, ça faisait partie de mes questions. Pourquoi le PDMS? Parce qu'il y avait des solutions alternatives avec des films secs de type époxy qui permettaient de au moins régler, faciliter ce problème d'alignement que tu as rencontré. Et sur une monocouche, en plus tu as des canalisations de 50 microns d'épaisseur. Les problèmes de contraintes et de craquelures ne sont pas très stringents. Voilà, c'est un commentaire. Comme ça, j'y reviendrai pas tout à l'heure.
Donc à ce niveau-là, donc au niveau de la fabrication, c'est vrai qu'on peut faire des choses avec des films. Nous, l'idée, c'est qu'on allait travailler avec une partie microfluidique et que comme on allait avoir une partie de PDMS pour la microfluidique, comme au début, j'ai commencé par essayer avec de la SU8. Et j'ai compris tout le challenge d'utiliser de la SU8 et toutes les contraintes que ça apportait. Et ce qu'on a essayé de faire, c'est de dire, ben voilà, en fait, une fois qu'on a fabriqué notre puce microfluidique en salle blanche, l'idée, c'est pas d'avoir à faire un travail supplémentaire quand on est en laboratoire. Donc ça, c'est vraiment cette idée que tout est fabriqué en salle blanche. Et la seule étape critique, c'est finalement l'alignement. Et si on arrive à faire cet alignement, la puce, on la sort de salle blanche et on peut l'utiliser. Alors qu'en SU8, ce qui était là avant que j'arrive, c'était qu'on venait faire, on va dire, fermer la puce juste avant l'expérience. Donc c'est quelque chose qui pouvait amener à des problèmes et qui finalement, on allait s'en rendre compte au moment de la manip. Donc là, c'était décaler le problème avant la manip pour avoir une puce packagée qui est prête à être utilisée. Donc ça, c'est un premier point. L'autre point sur tout ce qui est film, alors tout dépend de ce qu'on veut faire. Est-ce qu'on veut aussi utiliser le film pour le canal microfluidique ou si c'est juste pour fermer? Donc ça, c'est, on va dire, deux approches. Si on fait tout, on est plutôt limité au niveau de la résolution qu'on peut obtenir sur les films en ordile. On n'est pas au micron près.
Ouais, mais il n'y a pas que l'ordile.
Il n'y a pas que l'ordile, mais enfin, c'est juste pour élaborer sur un des points. Donc là, il y a aussi une littérature très grande où je ne suis pas rentré dans les détails. Je ne suis pas spécialiste des différents films qu'on pourrait utiliser. Et là après, si on vient refermer, mais qu'on a déjà un canal en SU8 ou en PTMS, l'idée c'était d'avoir tout en une pièce pour diminuer les étapes aussi qu'on allait avoir. Voilà pour quelques éléments de réponse à vos questions.
Oui, mais effectivement, il y a des alternatives possibles en fait.
C'est pas la seule chose.
Très bonne réponse.
Nous on s'est positionné de cette façon.
Alors, sur les mesures que tu présentes, on va aller directement au transparent 76 que tu as présenté sur les électrodes soit planaires, soit en vis-à-vis des... C'était le slide 76. Moi, il y a un gros point qui me gêne dans ce que tu montres, c'est qu'à aucun moment tu ne prends en compte la présence des électrodes et leur influence sur la mesure d'impédance.
Alors ça, c'est l'étape d'après. Avant même de prendre en compte l'influence des électrodes et tout ce qui peut se passer au niveau des capacités et de leur, on va dire, principalement à basse fréquence notamment, pour moi, c'était très important de comprendre déjà ce qui se passait sans les électrodes. Et c'est un champ dans lequel j'ai commencé à rentrer et qui est assez complexe au niveau rien que cette partie-là, comprendre quel va être le signal obtenu par une particule située entre deux électrodes. On va dire qu'arriver à théoriser ça et arriver à se rendre compte, déjà avec une particule centrée, au niveau de ma thèse, enfin, ça c'est une partie que je n'ai pas du tout abordée dans le manuscrit, mais c'est une partie que j'ai traitée en partie dans ma thèse, rien que la validité de cette formule-là, avec tout tout ce qui est transformation conforme, alors c'est pas quelque chose dont je voulais rentrer au niveau de la présentation, c'est déjà une...
Oui, mais c'est connu depuis, tu cites effectivement la transformation conforme des électrodes pour avoir une expression analytique du champ et en extraire...
Exactement.
C'est des choses qui sont connues dans la littérature, mais il faut aussi plonger, je suis d'accord. Mais le point clé...
Non seulement il faut s'y plonger, mais les formules, en tout du moins de ce qu'on a vu, la validité des formules n'est pas forcément donnée. Les théories derrière la démonstration des formules théoriques n'est pas donnée. Et ce que j'ai observé, c'est que les formules théoriques avaient des limites, fonctionnaient bien dans le cas de l'article qui a été publié, mais peu dans d'autres cas. Donc il y a tout un travail que je n'ai pas voulu détailler ici, mais que j'ai réalisé, et je peux vous en montrer un petit peu.
En fait, ma question, elle est vraiment, quand on va devoir prendre en compte les électrodes, comment on va pouvoir utiliser ces travaux, notamment dans le contexte MyDep. C'était une des questions qui... si moi je veux faire une mesure, est-ce que je vais trouver pareil que dans MyDep finalement
Alors MyDep n'est pas encore à l'étape mesure. Il manque justement cette influence des électrodes. Et l'étape suivante, ce serait d'ajouter la mesure d'impédance, déjà sans les électrodes et avec
Est-ce que tu as déjà une idée de dans quelle mesure ça va réduire les modèles ou complexifier la mesure?
En fait, ça va complexifier énormément. D'autant plus, et ça c'est ce que j'aborde légèrement ici, et dans la thèse, c'est que tous les modèles sont faits avec une particule centrée, dans le canal microfluidique. Dès qu'on commence à décentrer, c'est ce que je montre, le signal n'est plus du tout le même. Donc avant même de passer à l'étape des électrodes, si on n'a déjà pas le même signal pour la même particule, on a déjà une variabilité énorme et on ne pourra pas en tirer quelque chose.
Alors, donc là, il faut optimiser la fraction volumique, c'est ce qu'on pour avoir une mesure sensible, hein, c'est ce que tu as décrit avec les différents types de canaux entre deux électrodes. Ça revient quelque part à maximiser la fraction volumique puisque l'impédance est directement reliée à cette fraction volumique.
Au moins avoir une fraction volumique donnée, constante, à un certain endroit du canal.
Oui. Alors, est-ce que par rapport aux mesures que tu nous montres où il y a des variations, alors c'est là où j'ai pas bien fait le lien entre l'impédance et ce que tu exprimes qui est un blocage de courant.
La variation relative de courant, exactement.
Voilà. Est-ce qu'il peut y avoir un lien entre la variation que tu exprimes qui est inférieure au pourcent et les modèles sur lesquels tu t'appuies?
Alors là, le travail qui a été effectué, c'est uniquement des mesures finalement à basse fréquence où on est sensible au volume. La même cellule, du moment qu'on arrive à ne plus avoir cette influence de la hauteur, on pourrait commencer à travailler à plus haute fréquence et dans ces cas-là, on pourrait regarder les différents modèles. Mais là, finalement, c'est une sphère homogène qu'on place, une bille en polystyrène, et on regarde l'influence de ce volume-là. Ce qui d'ailleurs est une influence significative par rapport à l'autre, où on va commencer à avoir des résistances en parallèle. Là, c'est finalement uniquement le volume global qu'on va investiguer, moins le volume de la particule. Et si on commence à regarder avec des résistances en parallèle, on va dire que le signal, déjà, la variation de signal sera beaucoup plus faible. Donc, il faut être capable déjà de voir au niveau du volume avant d'aller plus loin.
Oui, d'accord. Donc, tu n'avais pas accès aux signaux d'origine, en fait?
Si, j'ai accès aux signaux, mais on a décidé de travailler à basse fréquence pour regarder le volume. On n'a pas voulu monter en fréquence.
D'accord. Ok. Bon, je vais m'arrêter là pour les questions, parce que j'en ai déjà fait pas mal. Je vais encore te remercier pour tes travaux et pour ton apport sur ce contexte interaction cellule-champ électrique qui est très très belle. Merci.
Merci Olivier, thank you very much. Hop, le micro dans ce sens-là. Professeur.
Thank you. Well, I would like to start by saying thank because I am very happy to be here in this invitation because it has been for me an opportunity to read very interesting work and that you have done very impressive because you pose a problem which is for me I am used more to fundamental problems. This is more closer to solving something really important like have been working in the lab, you have been doing theoretical analysis, modeling, numerical analysis. So for me, that's a complete piece of work, really very, very good. So, and now I would like to move on to some questions. Some of them will be maybe fundamentals, but yes, when you write the force, the dielectrophoretic force, you say that this is for an electric field applied which is stationary. And I would say that I think that you are saying that this is not a traveling wave or something.
Exactly. This is more in this.
But I would also point out the possibility that it's not only stationary, but that you have to, that the gradient of the phase should be zero. And that could be different from zero when you work near the relaxation frequency of your device. Okay? That means if you take into account the polarization of the electrodes, etc., you will have the impedance of your device will have a relaxation. If you work near the relaxation,
Yes.
then you will have, or maybe not, that depends on the shape of the electric field, imaginary and real parts, and then you may have the other term. Okay? So in order to have the dielectrophoretic force like we use normally, we should avoid some frequencies of the device. That's
Yes.
okay. That's one comment. Now, in the simulations, you use 2D, two-dimensional simulations. You say that the 3D will be, of course, much more consuming, etc. But what can you say about them? Because to me, the electric field will be more or less okay, because the electric field lines will be parallel to the top and bottom walls, because they are dielectric.
In fact, we did some 3D simulations. I didn't have time to put it completely in the manuscript, but we did them. And let's say the first part with using 2D simulation, it was more for understanding and to try to see what would be the optimal configuration. But when we moved to the 3D simulation, we saw that we should change the voltage and to change the speed, but it was at the end more like an adaptation to move from the 2D to the 3D. It was not changing completely the concept. So at the end, it was easy to perform all the optimization on the 2D and to see how we should adapt voltage and flow in the 3D configuration. That's more the approach we had.
All right. I understand that. And also, but in my mind, the electric field is probably okay. I mean, in the 2D to 3D, it shouldn't change much. But the 2D simulations of the fluid flow, to me, are more problematic, because you have the bottom and upper wall, and those are zero velocity constraints.
Yes.
So what?
So I mean, this is results I'm not presenting here, but we have done those simulations and we see that we have this parabolic profile in both dimensions, and that after the question is, should we simulate some gravity or should we not simulate gravity? But most of the time, because of the fluid, the particle is not completely at the bottom, so at least it has a significant velocity, and at the end, working in 2D or working as a cut plane in the 3D simulation was giving similar results. Just modulo the modification of the voltage.
Changing some.
Yeah, adapting the voltage, and that's also I was quite brief about it. At the end, it's all this concept of the liquid electrode.
Right.
That you go from this 2D configuration, coplanar electrode, and at the end, at some point, you have kind of an equivalent vertical electrode.
Yeah.
So at the end, if you work with the 2D electrode, the significant change that you have to go when you go from the 2D to 3D is only modifying the voltage at the end. Because the equivalent voltage applied is not the one at the electrode, it's a lower voltage depending on the access channel to the central.
Right. Yeah. Okay. Yeah, the liquid electrode has that.
That's why we did also mostly 2D. It's only a question after of adaptation of the voltage.
Okay. How can we interpret the figure 5.4, which is you have the position on the channel versus the radius of a spherical particle, I guess, and that is similar to the position of the cluster versus the number of bits.
The 5.4?
5.4. So they are very similar. To me it's surprising,
Yes.
but
So I don't have them in the presentation, but I have some additional simulation.
yeah, like that.
It's
for instance. So that can be used for the cluster. I mean,
Yes, that's indeed what we were showing in this figure. So in this figure, just for the general audience, we were comparing the evolution of the diameter of a particle and its trapping position. So we see as the position increase, in fact, the cell is going from being very close to the outlet to, for larger, let's say the maximum cell we have, being way closer to the inlet. And in this work, what we did is we looked at the polystyrene beads, and we saw that, in fact, the more cells, we can't say, I mean, the more polystyrene beads, we can't say aggregate because they are not permanent, but at least forming this cluster, it was interacting like an enormous dipole.
I see.
And in fact, the number of polystyrene beads composing this dipole was relatively similar to making a kind of equivalent size. If we compare it to the simulation, like two beads were not reacting like two individual beads, but it was more forming a larger dipole that was interacting with the field and push more towards the inlet.
I see. Yeah, but even so, a cluster which is like one after another, not like a column,
Yes.
will not have the, for instance, the same
Yes.
those drag force and sphere. So it's
Completely true. So it's not exactly you can't really translate from one to the other, but at least because of the flow, so always pushing particles, it was not forming a long chain. And as you see also in the video, cells, I will maybe re-show the video of the trapping with 8 electrodes. So if we look, you see you have two cells, and when the third cell arrives, it's not attractive. It's like moving a bit, like kind of push towards the limit, and then coming back to touch. You see, it's not just joining, it's not forming this chain, and that's a question that is also very fundamental for us, and why is it happening this way? You see, the cell is coming, and it's not like it's just joining and adding to the line. It's really going, and you see it's trying to kind of avoid this enormous cluster, and will be touched, and at the end will be kind of integrated. So let's say it's this similar shape is more valid for this one, because if we look back at the beginning, the final position, when it's larger, it's here, and its original position at the beginning were, in fact, a bit closer to the outlet. So we really see this effect as kind of making an equivalent larger particle.
I see. And now in this plot, let's say, if you reduce the radius, there will be a limit when the particle.
Yes.
Yes. Do you have that
And that,
estimate? Or
yes, I mean in my design, after it also depends on the voltage I apply and the speed, but at least for those particles, I mean this set of configuration, we saw that the minimum radius was 3 micrometer. And now if we look at the different forces, so in green we have the drag force, in blue we have the TEP force, and in black, dashed black, we have the resulting force. So we see that little by little, when the particle is too small, we have almost no equilibrium position. So if we have just a small variation of the flow, the particle will go. So below 3 micrometer, for this set of parameters, it was not possible to perform the trapping, at least in simulation.
No, yeah, I think the 3 microns makes sense because the force, the dielectrophoretic force is
Proportional to
power
R
3,
squared.
The
R
drag
cubed,
force is
you're
power 1, so in the end, the ratio between the two is power 2, so that means that it will be very strong value, I think. I mean, very robust. You probably cannot trap below 3 microns unless you use a lot of voltage.
And after the question is, it's always a balance. In fact, the conclusion of the PhD, it's always a trade-off between the drag force and the deep force. If you want to reduce the deep force, you need to reduce the drag force. But the limit is when can you still bring cells into the trapping chamber? Because if your cells are all like sedimenting at the inlet of your microfluidic chips, then you cannot form anything. So at the end, it's always this trade-off. And this technology might not work for too small particles because you would need at least some flow, but the drag force will be smaller when the particle gets smaller, and they will never reach the trapping chamber. And even if they reach the trapping chamber, you will need an enormous voltage to do it.
Right. Okay. Now, you said that you place additional outlet channels to reduce the velocity.
Yes.
But but the results that you show do not show any velocity in those channels. So don't you think that you should avoid just?
I think we should optimize them.
It could be deleted. Or not.
I mean, the thing is,
Or maybe bigger. Maybe make them bigger.
you are completely right. I mean, the influence, at least in simulation, is almost nothing. But experimentally, we saw that adding this additional outlet were in fact helping us to reduce the voltage used for trapping. This is something that I completely agree with you. When I read it back all the simulation, I saw almost nothing in the side out, in the auxiliary channels.
Exactly.
But that's why I say also as a conclusion that in fact, I think it's not optimal what we have done for those auxiliary outlets. Making them wider and playing more with fluidic resistance, we should also be able to improve the trapping. The limit is the fact that in this case, we wanted them relatively small. I will just show this slide. We wanted them relatively small because we wanted to avoid particles to go there.
Right.
The problem is if we make it as large as this one, particles will be able to go there, and maybe the trapping in fact won't work anymore because maybe they will prefer to go there. And here all the trapping is done to be kind of made by this electrode that is in the flow. And in fact, what we saw with the 8 electrode configuration is we need to have an electrode facing. Because originally I wanted to use only 2 side electrode. The force is way too small. So we have to have this electrode directly, let's say, longitudinally in the microchannel, and to use additional electrode to kind of plane, have the maximum resultant longitudinal.
Okay.
Did I answer your question?
This figure is complicated. The one of the figure 5.9, 5.10. Yeah, I have to try to understand it, especially the green and blue bits. Yeah, it's complicated.
Yes.
But now I think I understand. But at the beginning I didn't, because I said, why do you have blue inside the channel? There is no dielectrophoretic force inside the channel. But in fact, both are very small. No? The vertical force.
Yeah, it's almost zero.
Is both.
Yeah.
So it could be.
The trick was to try to represent when we have this equilibrium, which force is predominant in which part.
Exactly.
And that was something that was really difficult to get. And this is in fact a trick I use working with the elevation and make it proportional to the value of the force to try to represent which force is bigger in which part.
Because, for instance, the dielectrophoretic force, you can use the energy. I mean, it derives from a potential. But
Yes.
the problem is with the Stokes flow, I think, that you cannot do that because the profile of the energy is very useful to know where you are, right? But then the Stokes flow, I don't know what to do with that. Okay. ask you, of course, because I work on fluid flow, electrically induced. Have you seen electrically induced fluid flow?
I have. I read papers about it, but
What have you seen in your device?
I'm not sure.
You're not sure.
I think, let's say, coming back to the configuration with 8 electrodes, that we have a significant effect coming from the flow. But do we have some electroosmosis or some gradient of the conductivity? I'm not sure. Directly with the electrodes.
Yeah. You didn't use, let's say, small colloidal particles.
No,
just to see
we really limited ourselves to 8 micrometer.
Just to visualize the flow. Okay. In your simulations, you didn't, in the simulations of the impedance, okay, yes, in the 6th chapter, you didn't, it was very, for a frequency, but it didn't matter that frequency, right?
Not so much. We are just looking at the size.
Because for instance, if you want to do impedance spectroscopy of your cells or your clusters, you will need to include the capacitance, well, of the double layer of the electrodes, etc.
Yes.
I mean, the polarization of the electrodes and things like that.
Completely right. Similar answer to what I said with Olivier. In fact, we are not taking this into account because we realize that the height of the particle was influencing so much the signal that first we need to tackle that. Because the global picture is my DEP. We wanted to say all the work we did on impedance, we want to have the features and to say we want to predict what will be the impedance of a certain kind of cell in a specific configuration of the electrode. And the problem is like it's way more complicated than what we expected. Like the literature is not dense enough on all those conformal mapping in the microchannel, depending on if we have particles that are facing electrode, top and bottom, or coplanar. We are kind of missing an intermediary part, a mathematical part. And that's something I started to tackle. And also I have Gregory Vial, a professor of mathematics here. And we try to tackle it from a theoretical point of view and to see also how to address this volume fraction. Because before, even speaking about the electrode and the polarization, we need to integrate this volume fraction and to have a quantification of this volume fraction. Because right now, if we look, for a big suspension, when we have a certain number of cells, we can speak about a volume fraction if we have two facing electrodes in an enormous cuvette. But once we are starting to work in a microchannel, evaluating the volume fraction is very complicated. And as I said, in fact, the evolution of the signal between two electrodes, it means that we have, by changing the height, a variation of the volume fraction.
Right.
It's not seen the same way because the field lines are denser at the bottom than at the bottom of the microchannel. So that's also like to try to give a response to your question. Yes, we need to take into account the electrode, but the first step is to better understand this volume fraction and how to quantify it.
Right. But this will be, I mean, this is moving in the direction of doing experiment at different frequencies.
Yes.
Right.
Yes. This is, the design was.
For instance, if you want to know if your cell after electroporation process has been perforated membrane, you will need, I guess, to do a complete.
Or at least two frequencies, a lower and a higher frequency.
Exactly.
Yeah, I completely agree.
Okay. Well, in the outlook, you said that in the simulations there were more things needed to do, of course, like include dipole-dipole interaction. And if you want, if you like, the particles are not dipoles anymore. The clusters are so big that the gradient of the electric field you have.
This is only valid at when we are working with a single particle. When we are working with a larger cluster, it's the dipolar approximation is is gone.
Exactly. So maybe that's another direction. If it is maybe not very, it's more fundamental than experimental, because in the end, you have done the clusters using the
maybe we will, in fact, still miss something.
Maybe
Or
it's more fundamental
yeah.
than more theoretical. After what I have seen in your work. Well, yes. Did you use the ellipse in any time? I mean, the ellipsoid.
I found some literature with model in it,
But not in your.
but I never used it.
Okay. Yeah, it is just a comment. The point is that these formulas use confocal. I mean, they use some focus of different ellipsoid, and these do not keep constant the thickness.
The thickness of the membrane, I think we discussed.
So that means that you need to choose which part will be for you the thickness of your brain. But it's just a comment.
Yeah. To be theoretically valid, you are completely right. But it's more to try to apprehend what is happening when you have this membrane. So it's, let's say it's not something, in fact, you would see when you perform the experiment, but at least it gives you an understanding of what is happening when you add layers in this different shape. Let's say my DEP, right now it's not, you use my DEP and you expect to see exactly the same thing in the lab. It's not yet ready for that. It's more to try to understand more or less what you should see, and once you have the result, to try to understand how you should tune your model. So it's more like you start with my DEP, you go to the lab, you perform your experiment, and when you exploit your data, you are coming back to the model and say, maybe the model was not perfect, and what should I modify to make it fit better? It's more this concept of my DEP. It's to help you to go to the lab and to understand what you are doing.
To conclude, I think that your presentation has been very good, so I'm also very happy for that. And I think that there is a lot of work in the future based on this. I have enjoyed it.
Thank you, Antonio.
Thank you.
Ok, merci. Bon, je vais garder la parole quelques instants avant de passer la parole à tes encadrants. Hein. Alors je vais le faire en deux temps, je vais faire la première partie en français pour dire des généralités.
Non,
Ça marche? Pour dire quelques généralités, puis après je poserai 2 ou 3 petites questions en anglais. Alors d'abord je voulais remercier les encadrants de m'avoir invité, ça me fait plaisir d'être aujourd'hui, puis ça me fait plaisir de te revoir. J'ai remarqué que tu as fait beaucoup de progrès dans l'utilisation des logos, je vois que tu t'es professionnalisé sur ce point-là, c'est très bien. Voilà. Donc moi je trouvais, alors mon collègue d'à côté a dit exactement les mots que j'aurais employés. C'est-à-dire vraiment ce que j'ai trouvé autant dans ton manuscrit que dans ton exposé, c'est une manière d'aborder les choses qui est d'abord extrêmement posée, extrêmement rigoureuse et extrêmement carrée. Voilà, donc le terme carré, c'est... Voilà, j'aurais pas utilisé un autre terme que celui-là. Voilà, donc c'est un travail qui est, je trouve, très complet, très intéressant. Alors effectivement, moi aussi, j'ai eu la petite frustration de manquer un petit peu, mais d'ailleurs je vais poser une question sur ça tout de suite, sur la partie biologique, c'est-à-dire ce bel outil hein, que tu as fabriqué. À quoi on peut l'utiliser concrètement et qu'est-ce qu'on peut en attendre. Enfin, en tout cas, ce que je voulais faire en premier lieu, c'est te féliciter pour la qualité du travail que tu as accompli, pour la qualité du manuscrit qui est extrêmement bien fait, et aussi pour la qualité de ton exposé que j'ai trouvé particulièrement clair. Enfin, vraiment beaucoup de qualité pédagogique et aussi dans la richesse des éléments que tu as pu nous communiquer en 45 minutes, facilement 45 minutes. So now I have only 3 questions I would like to ask you. And the first one is about possible application. There is one that you mentioned, which is to use that kind of system to perform electroporation or to study electroporation. And what I would like to ask you is, what do you expect, what type of information do you expect from this kind of experiments? Because there are there have been many studies about electroporation. Electroporation is somewhat very mature because it is already used in clinical application or in veterinary application. So what are the kind of information you think we could expect from that kind of experiment and that we don't have yet?
Okay. So the first thing is with our design, we are able to create aggregates of controlled composition. We can decide how many cells we want in the aggregate. And that's something that is normally not really possible, or at least you can create aggregates, but of a very large size where we don't control the properties individually of the cells. And that's something we are bringing, or at least trying to implement in a full lab-on-chip system, where we can control the particles that are composing individually the aggregate after the general composition of the aggregate. And that's in fact coming to this slide. Once we have the aggregates of a controlled composition, we want to see in fact what should be the voltage that we should apply when we are modifying the size of the aggregate, how many cells we are using. And that's something that is still missing. We are either working at a single cell or at a specific size of aggregate or specific size. But when I say specific, it's not even in terms of number of cells, it's globally of size. It's something you put in culture for 3 days and you are using it. It's not something that is very precise up to the cell level. It's a general shape. Or the macro application is you have a body that is way more complex than a tissue, and of course it's used. But what the clinicians are really doing when they are performing electrochemotherapy or electroporation more generally is they have a set of parameters and they are in fact not using them. They are increasing of 10-20% the parameters. So when the theoretical aspect is coming back, you are, but why are you using more than what was required? As I said, because it's working. So in fact, this is all this understanding that we're still missing something. We are missing really this intermediary step from all the results that and the very mature literature that exists on single cell, and for the tissue and the body and our larger aggregates, where we have, let's say, a big ocean of results, and it's difficult to say why the results are when we see a discrepancy in the result. But that's one. It's more or less what I would like to answer to this point. It's trying to help to better understand how, when you go from a single cell to a larger aggregate, how the electroporation parameters are evolving. This is more the application in this case.
Okay. And the other part of my question was, so the idea is to create aggregates with different types of cells. So as trapping is depending on both the dielectric properties of the cell and the size of the cell, how will you work? How will it be possible to keep cell trapped in the trapping chamber and in the meantime bringing other type of cell to the aggregate and make them
touch each other?
Yes.
So I would say that the answer is yes, and the reason why, it's because we are working at the maximum of the closed-osmosis factor, of the real part of the closed-osmosis factor. We are working at minus 0.5, which is the same. So working with a quite conductive medium, but not too low in conductivity, allow us to have this force that is maximum. Of course, you may have some difference in the speed of the particles in the chamber, but once they start to be relatively close to each other, because at least the force is relatively similar, if we modulate only the radius, that will influence, in fact, quite strongly the force, we see that we have, in fact, very sharp DEP change. But it's appearing above in a very small frame. It's not from the inlet to the outlet. I want to find, again, this is it. You see how it's changing. It's dramatically changing over a very short distance. So this effect of the discrepancy in the trapping would happen in the micron or in the few microns distance. So we think that using, in fact, the fluid and the combination of the fluid, and in fact something that is not modeled in the simulation, in the particle is influencing also the fluid. And that's something we don't have in the simulation. We are just macroscopically observing the global effect. but without taking into account that the particle here, imagine if it's here, it should modify the flow line. So we really believe that it's something that would be possible in our microfluidic chip.
Okay, thank you. And I have a small question to finish with, is that you mentioned that when you fabricate the devices, that you have to align very precisely the fluidic part over the electrodes. Okay, that's it. And my question is, what is the influence of misalignment on the performance of your trapping device? Or, in other words, what is the tolerance of misalignment you can afford?
So, I did this study for impedance. Mm-hmm. I saw that, in fact, depending on the design, you can be quite immune to misalignment. But for the DEP, the problem is at least you need to have the electrode everywhere in the microchannel. Like here, if you have the electrode only on only a portion, you will use an enormous amount of force. The problem is after playing with the distances, and that's the work that we didn't do. We didn't study in details how the misalignment would influence the position of the trapping, or if we can do the trapping at all. So I don't have a precise answer, or more precise. I can just say that in this case, even the shrinkage might, okay, I want to elaborate a bit more. The trapping that we perform in this design, like this, where we don't have any tolerance possible, because we had 50 and 50. So after we understood the kind of modification we need to make, and we made it 60 micrometer. The problem is, like in this design with 8 electrodes, any misalignment you will see it directly. But with the design with the 4 electrode, to find and to understand the misalignment is very tricky. Even more that you align on a precise location. And as a, let's say, practical aspect, I have a general alignment on the microfluidic chip, on the corners, and I perform the very fine tuning directly in the zone of interest. Because otherwise I can't have such good alignment directly at the microfluidic, at the full microfluidic.
And you think you need a 1 micron alignment precision, or is it?
So the question is above which distance? And so the result is taking into account that you still have some shrinkage. So the shrinkage is not completely compensated. So over, I think what I wrote in the manuscript is 3 millimeter. I could quantify the misalignment. But you have also to decompose what is due to the misalignment and what is still due to some remaining shrinkage. So I would say that it's working at a smaller scale, but if you want to work at a large scale, you need to characterize way more precisely before saying you have a 1 micrometer precision.
Thank you very much. That's all for me. So, je vais passer la parole à François, Marie, et on finira par Philippe, peut-être. Marie de peu importe.
So I won't ask you any questions, but I think I will use the spare time for a few acknowledgements. So first, I would like to thank all the jury members for having accepted to be part of this thesis committee. I think the questions that raised were very interesting, and they will be helpful for future work. And I want to say that I am very grateful to Professor Philippe Renaud for having accepted this collaboration, and it was also for providing you such an excellent environment for your thesis. So it was very interesting for us, and I really hope that we will have some chances to still collaborate in the future. And I think I should also thank Harald von Lintel. I don't know if he's here today, but I know that he has also
on YouTube, yes,
been a great support for you at EPFL, and also my colleagues François Buret and Olivier Fabreg, because you strongly interacted with them also. And I am very grateful to Riccardo Scoretti, who is the coordinator of the Dynamo project dealing with electroporation modeling at the multiscale. So I think it's with this project we have a very interesting subject here to tackle, and so also I thank Julien Marchalot, who collaborates on this project. And now, if you don't mind, I would like to switch to French, maybe to express my thanks to Jonathan, so that everyone can understand, in particular in his family. Alors, donc, Jonathan, je voulais te dire que c'est assez spécial pour moi là, de réaliser que tu arrives à la fin de ta thèse. J'ai pas vu le temps passer, et pourtant je réalise que finalement on se connaît depuis pas mal de temps, puisque tu avais pris soin de prendre contact déjà 2 ans avant ta thèse, puisqu'avec le sens de l'organisation qui te caractérise, tu as vraiment essayé de choisir à l'avance ton sujet. Donc je dois dire que j'ai été ravie de te voir revenir après tes 2 ans de master, avec surtout dans tes valises une collaboration très très intéressante avec le laboratoire de Philippe, dont je suivais les travaux déjà depuis quelques années. Et tu as très très bien suggéré cette collaboration en étant le plus souvent en Suisse, hein, c'est vrai, on ne l'a pas dit, mais tu as passé le plus clair de ton temps à l'EPFL. Et pourtant, je n'ai pas eu l'impression que tu étais plus loin que les autres doctorants de l'équipe, puisqu'on a quand même réellement pu rester... j'ai trouvé que la collaboration, elle était très riche et très active, et qu'on a pu beaucoup échanger pendant toute ta thèse. Donc ça veut dire que tu as très bien suggéré ça, et prendre le meilleur de chaque laboratoire et pour arriver à construire au mieux ton projet. Je crois qu'il y a déjà pas mal de choses qui ont été dites sur ta rigueur, ta façon de travailler, ton sens de l'organisation, mais je voudrais aussi dire ta ténacité et puis ta capacité à faire avancer un dossier. Là, tu m'as vraiment impressionné, je dois dire, alors que ce soit parce que tu constates que le PDMS n'est pas plat ou rétréci. Alors, il y en a beaucoup qui auraient misé sur la statistique pour dire je vais peut-être en avoir 1 sur 10 qui va être bien et je vais me débrouiller avec celui-là. Toi, pas du tout. Tu cherches à résoudre les problèmes les uns après les autres. Tu t'y attaques et tu arrives à cocher la petite case, à passer à la suivante. Que ce soit donc pour ce PDMS plat ou pour aller poursuivre un auteur qui a écrit un article il y a 10 ans où il y avait peut-être une petite coquille et tu voudrais bien qu'il t'explique pourquoi. Donc vraiment très tenace, et je pense que c'est vraiment un de tes points forts, hein, parmi tant d'autres. Mais donc au final, je ne vais pas monopoliser la parole trop longtemps, mais voilà, je voudrais vraiment dire que tu as accompli beaucoup de travail dont tu peux vraiment être fier. Je trouve que ta présentation d'aujourd'hui montrait vraiment que tu as fait un très beau travail. Et donc moi je suis très fière d'avoir pu participer à ton encadrement. Et donc je te remercie et je te souhaite le meilleur pour le futur.
Merci Marie.
Merci. Je vais remercier tous les membres du jury. Marie l'a déjà fait, mais je m'associe pleinement à elle. C'est toujours des journées un peu longues et difficiles pour certains avec les moyens de transport. Donc, effectivement, c'est une thèse pour moi, donc je garderai un bon souvenir. C'est pas ma dernière, c'est dommage, j'aurais bien aimé m'arrêter là-dessus. J'en ai vécu des plus difficiles, nettement. Donc j'ai bien apprécié effectivement ta quantité de travail, et la quantité et la qualité du travail que tu as fourni. J'ai eu un peu peur quand tu t'es lancé dans MyDep. Et finalement, ça s'est très bien passé, parce que je me suis dit, ce n'est pas le sujet principal de la thèse. J'ai dit, oulala, je sais ce que c'est qu'écrire du logiciel, on va y passer du temps. Et puis, voilà, à côté, il y avait un autre sujet que Marie avait défini, et que j'avais à peu près compris. Et je me suis dit, on va... Mais bon, tu t'en es très bien sorti, donc félicitations là-dessus. Tu as travaillé dans un environnement, je pense, qui était intéressant, mais difficile, parce que tu avais des trajets à faire. Donc intéressant parce que tu as travaillé avec des gens qui n'avaient pas du tout le même background scientifique. Ça t'a demandé probablement des efforts pour expliquer, mais tu as des talents pédagogiques. Tu as pris du temps pour m'expliquer ce que tu faisais, c'était déjà bien. bon, tes talents pédagogiques, je dirais simplement un mot, c'est que tu as fait de l'enseignement à l'école et que ça s'est très bien passé, que j'ai eu des retours plus que positifs. Donc ça c'est très bien pour quelqu'un qui a une formation académique et qui a priori pourrait se destiner à l'enseignement. Donc c'est parfait de ce côté-là. Moi je vais terminer par une boutade. Je dis, bon, tu as réussi déjà à me faire comprendre ce que tu faisais, ce qui est bien. J'ai compris aussi que tu avais réussi à faire travailler un polytechnicien, ce qui est encore mieux. C'est pas facile.
Alexandre, si tu nous écoutes, c'est une petite dédicace pour toi.
En France, on tape toujours un peu un moment sur un polytechnicien, c'est traditionnel, je ne vois pas pourquoi on y échapperait. Donc ça montre que déjà tu es capable d'entraîner un certain nombre de personnes sur ce que tu as fait. Moi j'ai apprécié fortement ta démarche où effectivement on a l'impression que, je qualifierais de bulldozer parce que c'est peut-être un peu, enfin c'est peut-être pas très flatteur, mais je dirais c'est quand même très bulldozer. Quand tu avances quelque part, ben, on voit qu'au bout d'un moment ça arrive. Tu arrives où tu voulais aller. Quoi. Donc de ce côté-là, bravo. Et bonne continuation. Je ne sais pas ce que tu feras plus tard. On en a beaucoup parlé. C'est quelque chose de difficile à décider, mais je te souhaite avoir une vie intéressante.
Merci François.
Philippe, c'est à toi que revient le mot de la fin.
The mot de la fin, okay. Maybe I conclude in English so that we can share. So first of all, I have no questions because we had already many questions. So I only thing which is left is to congratulate you for the thesis and to express my just honor and pride to have you as a doctoral student and also the nice collaboration we had. So and it's also at the beginning was not clear how it could be handled and keep the working on both sides. And in fact, the impact of the collaboration was really great because you've done on the French side things that we were totally out of the business, and so that makes this thesis extremely good and wide. And you defend yourself quite well to these questions. Okay, so you're strong. That's very good, and that was really a pleasure to work with you. And I'd like to also thank you, of course, the co-director, for really hosting you and really also helping you, because it's really easy in such collaboration to lose the contact on one or the other side, and I'm very honored also by the jury, by the quality of the people around us. So thank you and congratulations.
Thank you, Philippe.
Merci beaucoup. Donc on va se retirer pour délibérer et nous revenons dans quelques instants. On verra si on attribue le doctorat. On va réfléchir. Ben, la photo, on la fera, hein? Ah, pour le stream. Il faut venir sur l'estrade pour... The jury members are requested on the stage. Okay. So, Jonathan, I will do it in French. Okay. Donc, Jonathan, le jury s'est réuni, a délibéré longuement et a décidé de t'attribuer le diplôme de docteur de l'Université de Lyon. Alors, comme tu le sais certainement, nous ne pouvons plus attribuer officiellement les félicitations, mais ceci dit, le jury, étant donné la qualité de ton travail, la qualité de ta présentation, a souhaité te délivrer des félicitations à titre oral. Donc, vraiment, très sincèrement, nous te félicitons de ton travail et nous te souhaitons tous une excellente continuation dans une carrière qui sera peut-être académique. Je ne sais pas ce que tu as l'intention de faire maintenant. Donc, bravo.
My turn. So I would like to thank all the members of the jury for coming here and for their comments. Sometimes quite tough, but I think it's giving a lot of quality to the work that has been performed. So thank you all for taking the time to come and to evaluate this work and to ask questions on it. So it's also my great pleasure to have all of you here at École Centrale de Lyon and also on live in streaming in different countries. I would also to mention all the people at EPFL from the LMIS4 groups that are attending the presentation. So thanks to everyone who is attending either here or in streaming. I would like to thank especially the members of the two groups that I've been working in. So Philip, thank you very much for your support, for letting me do as I wanted, in fact, organizing between the two different labs. Thank you for all the support you provided me for making fabrication in the clean room, for supervising students and performing experiments in the lab. And I'm deeply grateful for all the time I spent in LMIS4. Marie, François, thank you very much for you both. I mean, as you said, Marie, it's a long time ago that I came to you. It was in, if I am correct, 2012, December 2012, that I came to you when I was looking for a master that I wanted to do abroad. And we had some very interesting discussion. And I, at the end, decided to go to EPFL, and we always stayed in contact during this time. And it was really a great pleasure to interact with you. And basically, all the discussion we had before even starting the PhD showed me that it was the right place to be and to collaborate with you. So it was the beginning, and I think we continue for a long time together during the PhD journey. So it was tough, as you said. Also, it's something that we never mentioned here today, but we originally wanted to set a co-tutelle, and it was sometimes difficult. But at the end, I think with what we came with the organization I had, was a very nice place to be, and we could really perform all the best. So thank you very much, François and Marie, for all your time, all your answers to my email, sometimes very late at night, and all the discussion, even in the weekend, and also all the personal aspect that we shared together. And also I would like to thank all my colleagues here, all my, and at EPFL, all my students, especially Alexandre Keran and Soufiane Lasli, who did a really huge experimental work, so thank you to both of them. And now I would like also to thank all the people who came here, like all my former professors, mostly all the members of the lab here who came. And now I would like to thank my family a lot. Merci beaucoup à vous. toujours avoir été là. Alors, c'est pas quelque chose de facile. Quand on a quelqu'un qui a des grandes ambitions et qui veut aller un peu partout dans le monde, qui travaille comme un fou et d'arriver à suivre derrière. Donc, merci à vous de m'avoir donné ce support. Donc, merci à mes parents, merci à ma petite sœur et puis merci à toute ma famille pour toujours avoir été là quand j'avais besoin d'elle. Merci à tous mes amis qui sont là aujourd'hui et qui sont peut-être loin. Et aussi merci beaucoup à Irina pour ces derniers temps, pour tout le support que tu m'as apporté et toute l'aide que tu fournis aujourd'hui en permettant le streaming notamment. Merci à vous tous d'être venus et puis à très bientôt.